morning, everyone. If we can have folks grab their cup of coffee and sit down, we're going to get started. Yeah, we had a really successful day yesterday and a lot of fun with the presentations, and the poster night went really well last night. So we're into day two and a half of camp, and we've got another full day of presentations. This morning we have the dynamic methods session that Julie Bonnie will be chairing, and then about mid-morning we roll into our community and monitoring session, and both of them have some awesome lineups of presentations. I wanted to remind everybody, if you didn't hear the announcement yesterday, we CGRAT, everything we do, we always look to do better, and we rely on people's feedback and evaluations to let us know how things can be improved. And we have, we're trying a new format this year. We've traditionally done this online, and like any online survey, we recognize that they're hard to do sometimes, and nobody really likes them. So we're going back to paper format, and we have an evaluation sheet out on the registration desk as a little incentive to fill this out. When you fill this out, and it's all trust-based, because we don't have that desk man to nonstop, please grab yourself a raffle ticket. There's a two-piece to this, and because we want to give you the option of filling this out anonymously, so you can really put down your thoughts and comments. We won't know who you are. You can select to put your name on this, but if you want to fill it out anonymously, then just please add your name to the ticket and put it into the fishbowl. So we're going to be doing a drawing at the end of the symposium and giving away a couple of really nice CGRAT books. So if you can help spread the word, we had these available midday yesterday, so some folks are in and out. So we encourage ongoing sign-in. If you've already signed in, that's fine. If you see residents or community members that are coming in, encourage them to sign. We just want to get a head count. We really do look for the feedback, and we're constantly trying to improve camps. So so far, so good. We're doing great. I have just a couple more quick announcements. We have the Elliptic Museum tour happening. You probably want to coordinate that with Patrick Saltonstall. There will be lunch provided for the participants that go there. Hopefully not everybody. There is lunch provided, recognizing that anybody that's participating in the tour won't have time to grab lunch. So we'll stick to the same form yesterday, that we'll break for lunch and be back here at 1:40. So please coordinate with Patrick, and if anybody is like me and can't walk to the Elliptic Museum, maybe you can coordinate rides. I would even volunteer my car for folks that can't walk like I can't. And also tonight is the KSNSC reception. Quentin Fong is busy right now cooking a lot of food. If you've ever been over to one of our receptions, he puts out awesome plate of food out. So it's just a social event. Last night we had posters and little kids corner here. Tonight's just merely social and hanging out. It's open to the public, so please let people know that they can come over. There's usually lots of food. And then I just want to remind everybody too that Robin Cassidy has been organizing a science march. We have flyers out on the desk out here, and that's Saturday at 10 a.m. And I realize it's a busy day, Saturday, but I encourage folks to check into that and show your support for science and also marine science in our region. Okay, I'm going to turn it over to Session Chair Julie Bonnie. Julie Bonnie is with the Alaska Groundfish Data Bank.
Well, it's nice to follow another Julie. And this is session 3, Dynamic Methods. And I get the pleasure of introducing 6 wonderful folks that volunteered to make a presentation today. So the first person is Daniel Urban with National Marine Fisheries Service, and he's making a presentation on the molting process in golden king crab.
Thank you, Julie. Glad I fell into the wonderful person category. My name's Dan Urban. I work for Fisheries Service. I had also worked for the Alaska Department of Fish and Game here in Kodiak for quite a number of years. And you don't see Andy here, but Andy Nelk is Andy is here. Did you say anything, Andy? Andy is a co-author on this presentation. We're lucky over at the lab that we have the ability to keep and hold crab for extended periods of time, and that lets us, do some pretty interesting experimental work up close and personal. So it's a little different than working with crab that are out in the wild, but it still gives us a chance to do some pretty interesting things. The golden crab that we're talking about the molting in today, we brought into the lab for actually some tagging experiments, which were pretty interesting, and that could be the topic of a whole other talk sometime. But since we were holding them, we weren't really anticipating that they were going to start molting, but when they started molting, they gave us really a good opportunity to videotape them and make some observations. The golden crab are distributed from central British Columbia all the way across the northern Pacific to Japan. And this map here shows the locations where golden crab have been seen on the National Fisheries Service surveys over from '83 to 2016. And in that period, there were about 40,000 hulls that had golden crab in them. So you can see that they're quite widespread, and they tend to go out to be found along the 1,000-meter depth contour. But they may be deeper too. The surveys only generally go out to 1,000, so this could be a little skewed. And you can see that they also you can't see it now, though. Extend down to southeast Alaska. There's some fisheries in there, and also up in Prince William Sound and interior waters of southeast. There's golden crab found in those areas as well. The golden crab fishery is concentrated out here in the central and western Aleutian Islands, but there's a scattering of fisheries catches that occur along the shelf break in the Bering Sea, and then also along that fathom line here. So the fishery reflects the survey distribution. And there's quite a few, there's 130 of these blocks that couldn't be displayed up here because they're confidential catches. So that kind of demonstrates that it's a low-level fishery in a lot of areas of the distribution. To put the fishery related to some of the other fisheries that go on in the state, these are some from 2014. So you can see in the Bering Sea Aleutians, crab is only a relatively small piece of the pie out there, where pollock and salmon really dominate. So this would be the Bristol Bay salmon here, which is showing up. But crab is still, fairly large. And then if you look at just this chunk to the crab themselves, snow crab is the dominant species, and Bering Sea tanner. This is by value again in 2014, and Bristol Bay red crab. So the Aleutians are still a sizable piece of the pie, and of course those numbers will jump around depending on what the quota is doing in a particular year. Getting into some of the taxonomy, golden king crab are members of the phylum Arthropoda, and arthropods are a large and very ancient phylum. There are 3 times more species of arthropods than all the other phylums combined. So it just gives you an idea. They've had a long time to evolve and speciate, and they have a lot of very specialized adaptations, so they can fly and make spider webs and swim and burrow around and sting you and stuff. What characterizes them as arthropods is they have a hard external shell made out of chitin, and then they also have segmented bodies. So the king crab are in the crustacean class of arthropods, and they that class contains a lot of the familiar commercial species in Alaska with the various different shrimp with the pink shrimp side stripes and spots. And then, of course, we got our snow crab and tanner crab. And then there's the king crab group, which the golden king crab are part of. Talking generally about the molt cycle in crustaceans, this has been studied for a long time, ever since the early 20th century, and it's been divided into 4 major stages. Although you have to realize that it's really a continuum, and one sort of leads to the next one. So first we'll have the pre-molt stage, and I'll relate this more to how the visible signs in the golden crab. And in the golden crab, in the pre-molt stage, the abdomen starts to swell up, and also there's a lot of things going on internally, but the epidermis begins to separate from the shell, and the crab starts to take nutrients out of the shell and reabsorb them for use in the future. And you can see here that in this is sort of like a year in the life of an adult crab. So the molting period is really very brief compared to these other sections or stages in the crab. And then in the post-molt, the shell begins to harden up, and there's some other things going on internally. And then in the intermolt, that's just when the crab is out there eating crab. Okay, so you might want to take notes on this equation. This is how they determine like the number of legal crab in any given year based on last year's crab numbers. But the thing to look at here is the molting factor enters into the equation twice. And I was talking to some of the assessment authors, and they said that the proportion of the crab that molt is a really important thing. And both the pre-molt population trying to figure out how many crab are going to molt to legal size, and also the portion of molt, those are both poorly understood and partially. So that's why we want to understand the molting process better, because it'll help us answer some of these questions that are needed to set the harvest guidelines. The golden king crab, most of the literature says that they're asynchronous molting. They could have asynchronous molting, which would mean that there hasn't been any real observed period when the crab molts. So I think a lot of people realize that, say Dungeness crab, there's a certain time of the year when all the adults molt, the shore will be covered with shells, and tanners and other king crab do that as well. But it had been considered that golden king crab, based on survey information, that they were asynchronous, meaning they molted pretty much steadily over the course of the year based on the female reproductive, the state of the eggs and reproductive state that the females were in. When we held these crab, so we got 60 crab in to the lab over across the bridge, and held them in the tank, and we had them from May of 2014 all the way through to November of 2016. And so there was no molting activity. Then all of a sudden there was a huge jump in molting over pretty much a 2-week period, and then it just tapered up, it's ended again. So there seemed to be some sort of a molting period that we saw in the lab. And Dr. Foy asked me, well, do we know that wasn't just lab effects, or what exactly was going on? And I But it certainly was an interesting phenomenon, and it's certainly worth further investigation. Okay, now we're going to make this video play. I think. With help. So this is just sort of a brief segment of this crab and the molting process. And so there you can see the swollen abdomen, and the suture has split along the back of the shell here. It's starting to pull out. You can see the legs forming here. Here's the old gills. Oops, here's the old gills. Here are the new gills forming. It's edging its way out, and that must feel good. And so that's just a brief snippet of golden king crab molting, and that's a pretty general process how the crab split that back suture and back out of their shells for most of the crab. An interesting thing to note is the people who know crab, they have a very large claw, and then it has to be pulled through a fairly small portion of the rest of the leg. So you've got the large claw, and then it's got to go through these narrow areas, and that's one of the reasons why the claw meat is kind of a little strange, different than the rest of the leg meat, so that it can be retracted through that stricture in the leg. And that's it. I'd like to thank the crew at Atlantico that brought the crab in for us, and the Alaska Department of Fish and Game certainly helped maintain the crab down there in the lab, and Andy helped set up the video equipment, and also Liam staff helped maintain the crab and feed them and everything over the crab. So that's it. Andy, did you want to add anything about how it was set up, or if there's any questions about how we actually got that video? Andy, you could answer those. Thank you.
We're going to open it up for questions. So is there any questions? Right here.
You showed they molted synchronously in one May, but not the following May and not the first May. Have you got some idea what the environmental clue is that set them off?
No. No, I don't know if it's the plankton or daylight or just what it is and why there was that difference. And I think the lab setting is pretty artificial. They're getting regular food, and the water temperature is different than what they would experience in the wild. So there's a lot of things we haven't really teased out as to that pattern.
So they're on independent flows, so they wouldn't have been getting any chemical signals from other crabs that were molting?
Other than
independent flows and into the drain?
Other than what was coming out of Trident Basin, unless they got cues from other crab could have, one crab molting could have, cued other crabs at its time. I don't know.
Oliver?
Yeah, the new shell comes out of the old shell, so it's got to be slightly smaller. But the expansion of the molt must occur just after they pull out. Is it
Exactly.
rubbery? How do they expand their shells?
Well, they fill themselves up with water, and that expands the shell when it's still soft, and that's how the expansion takes place. And there's a lot of things going on at that point. But yeah, when it's still soft, they pump up. But then in 2 weeks, the shell's pretty hard, and they've done all the growing they're going to do for that go around.
And Julie?
How much do they grow in one mold, like roughly? Is it consistent, like a certain
Well, it seems like it's a 15% gain per mold, if I remember my numbers correctly. So, yeah, I'd have to really get back to the data to give you some exact numbers.
Ola?
Do we have time to watch that video again?
Yeah, I think we do. We'd be very comfortable.
Just pretty interesting.
That's pretty cool.
Catch
Sean?
Yep, so I know some crustaceans will eat their molted shell afterwards to try to recapture some of the nutrients and minerals. Did you observe any of that behavior?
We didn't see that, because they were on such a deluxe diet there at the lab. They didn't feel the need. Okay, so you see how swollen the abdomen is? It's not, there he moves, there's what's moving on here. This is a PC talking to a Mac. Okay, there we go. Or Mac talking to a PC presentation. Yeah, it's fascinating to see. And this whole process took, I mean, obviously more than 50 seconds. Was it about 2 or 3 days? I can't remember. When they were actually pulling out of the shell, that was about a 14 or 15 minute process.
He looks her age, or she does.
He does.
He does.
We've got hours more footage, so if anybody wants to come over to the office, certainly have a look.
So we have time for one more question if somebody else has another question. Over here.
Gordon.
Yeah, so I think the perception's been that golden king crab can molt pretty much any month of the year, and on average, their meat yield is less than red king crab because of that. But you're showing at least over a 2-week period some synchrony. Do you think it's kind of localized, so animals in one area might be doing the same thing, but over a broader area they could do it any month of the year? Anyhow, can you reconcile your observations with the perception?
I like that idea of localized, synchronized molting, and the habitat of the goldens and the evolutions is so diverse that different things are going on at different times. That makes sense, but I'm not sure.
Thank you. I got my cheat sheet here. Okay, so next is Christina Conrad, and hers is, it's complicated, the reproductive biology of the shortrekker rockfish in Alaska. Now, shortrekkers are a lot more beautiful than dogs, so, Mr. Trump, it's complicated.
So today I'm going to talk about the unique biology of rockfish. I'm going to introduce you to the shortrekker rockfish and present a little bit of data on a project that I'm working on looking at temporal differences in the reproductive biology of this species in the Gulf of Alaska. Rockfish of the genus Sebastes have a unique life history that includes internal fertilization and the live birth of planktonic larvae. There is some evidence that they are truly viviparous, which means that there is an additional maternal energetic contribution to the development of the larvae beyond the initial yolk investment. And in this region, they tend to have very seasonal, synchronous annual cycles. Parturition of larvae generally occurs in the spring to early summer months. And they have a bunch of they have complex reproductive cycles that include potentially a bunch of different phenomena. I'm going to concentrate on the first three predominantly, but mention all of the rest as well. So an abortive mature fish is an immature fish in which reproductive development is initiated, but is later resorbed. And the standard practice is to consider these fish immature because they're not contributing to any reproductive productivity during the year that they were captured, and they haven't in the past. And this is pretty common and has been documented in several rockfish species in this region. Skip spawner, on the other hand, is a mature fish that spawning season. And we've recently documented this in rough-eye, black-spotted, and short-wicker rockfish. When we talk about maternal effects, we're referring to the phenomenon where larger or older individuals within a population contribute disproportionately larger amounts of reproductive output. And there's a number of ways that they can do this. They may just have more embryos. They may have better embryos. They may either be bigger or have bigger yolk reserves. In some species, the larger females are capable of reproducing earlier in the spawning season, which may be advantageous. And I'm going to talk a little bit today about whether or not, when we look at skip spawning, whether or not larger individuals are more or less likely to do that. A few other weird things that rockfish reproduction, that we see in rockfish reproduction. One is that they can have a protracted adolescence, and this just means that they can start the early developmental process, and that this can be protracted for several years. It's possible that they may have fecundity down regulation. This just means that a portion of their development is resorbed, and that results in fewer larvae being released. And I'm not going to talk too much about partial total non-fertilization, something that I see in northern and dusky rockfish quite a bit, but I haven't seen it in these deep water species. That relates to the next one of parasites. These rockfish, all the rockfish that I study have some proportion of individuals that have these large copepod parasites. And I think that actually probably relates to some of these non-fertilization problems. But again, they're pretty rare in the deep water rockfish, so I'm mentioning here, but I'm not going to talk about it anymore today. So a lot of my research right now is focused on how do reproductive parameters vary. If we go out into an area and sample during a year, how static are the reproductive parameter estimates that we come up with, and how often do we need to reevaluate reproductive parameters, and how closely should our spatial coverage match that of either the fishery or the population. But the piece that I'm mostly working on right now is the temporal piece. So we're doing a study right now looking at variability in reproductive parameters of deep water rockfish. I'm going to talk about short-wickers today. And in order to do this, we collected samples in the 2009-2010 winter and the 2015-2016 winter. I'm going to refer to these as the 2010 and 2016 reproductive years because that's when parturition would have occurred. And today I'm going to present data where we looked at comparing estimates of length at 50% maturity, rates of abortive maturity, and rates of skip spawning, and also the maternal effect on rates of skip spawning, and comparing those between these 2 years. So the short-wicker rockfish is the largest of the Sebastes. It reaches a size of about 120 centimeters, which is about 47 inches, so just under 4 feet long, and about 40 pounds. They are a deep water species, predominantly concentrated right on the shelf edge continental slope. These are some catches from our biennial trawl surveys, and you can see that they predominantly occur right where the bathymetry tightens up on the slope. They are wide-ranging in the North Pacific Ocean with a distribution that is centered in Alaska, and they are found in all regions of the Gulf of Alaska. And there's no directed commercial fishery for this species. But they are caught as bycatch and trawl in longline fisheries. And these are the areas we sampled during those 2 winter periods. The purple stars are the 2009-2010 winter, and the blue stars are the fish we collected or the locations of where the fish we collected in 2015. And when we look at the length at 50% maturity, we found virtually no difference between the two time periods. The L50 was right around 49 centimeters, and the curves are very similar between the two periods. So, brief introduction to what abortive maturity looks like. I should go back a step and say when we assess maturity, we preserve the ovaries, and then we take a section of them, embed it in paraffin, take a thin section, and we stain it with hematoxin and eosin, which makes it these pretty pink and purple colors. And during the developmental period that we were looking at, alpha-atritic abortive matures should have a high rate of alpha-atresia, which is the beginning stage of atresia. And this slide shows a regular developing oocyte and 3 alpha-atritic oocytes. And the differences that you see is that the nucleus has dissolved, the yolk is starting to disintegrate, so it's no longer in these nice spherical shape, and the granulosal cells have gotten larger. So when we take a larger view of that, we see a bunch of alpha-atritic cells, sort of our baseline early development. These oocytes won't develop. And then there may be a few also still regular developing cells, but for the most part, this section is dominated by cells that are degenerating. And this wasn't terribly common in either year. There were less than 10 samples. The proportion was higher in 2016. This wasn't terribly unusual, about what we would expect. For skip spawners, what we are looking for to identify a skip spawner are later stages of atresia that are evidence of a prior spawning. So we're either looking for beta-atritic structures, which are sort of more compact versions of the alpha-atritic structures, which you can see there are 4 of those in this slide. Delta atresia is unique looking because the granulosa cells have this orange and brown pigment. And then we're also looking for post-ovulatory follicles, which are usually you can see some sort of a lumen within the slide. And skip spawning generally takes 2 forms. One in which there is no development at all during the reproductive years, so we're just looking for these little pieces of old atresia and evidence of a prior spawning. But we also have the case where they start to develop and the development is resorbed. And this looks a lot like an abortive mature fish, but again, we're still seeing these little pieces of older atresia that indicate a prior spawning. And we found during the first sampling period a high rate of skip spawning, around 60%. And during the second sampling period, also a high rate, but lower than the first period at about 43%. The more interesting part, or one of the interesting things that we saw, is when we look at skip spawning rates and plot them versus different size bins, we do see a bit of a maternal effect where there appears to be a size at which they are much more likely to spawn, and that is around 75 centimeters. So during the first sampling season, or the second sampling season, these younger fish were having more success than the first sampling season, and that's true for the larger fish as well, but overall there's just, it seems like once they hit a certain size, spawning is much more likely to be completed successfully. So just to sum all that up, our L50 was very similar. Abortive maturity rates were higher in 2016. There was a difference in the skip spawning rates of about 17% between the 2 years, and in both years, large fish were pretty successful spawners. So what do these results mean when we try and put them in a management context? Unfortunately, it's not really a simple question to answer, and this is really maybe more accurately a list of things that are sort of related to management implications. So they're not the target species of a commercial fishery, as I mentioned previously. And for short rickers, the catch limits that are established, or I should say, the catches for short ricker tend to be quite a bit lower than the catch limits for short rickers, particularly in the central and eastern region, which is where most of the catch is allocated. So we're really not coming up close to those catch limits on most years. The other thing is they're fairly data poor species. We don't production age them, so they are a tier 5 species, which means that their catch limits are derived basically only on 2 pieces of information. One is their national mortality rate, and the other one is biomass that's estimated in trawl surveys, in our biennial trawl surveys. So there's not really an easy way that this reproductive information right now fits into the management strategy. And sort of the big shiny new piece of information that has come out of the study are just the thing that we hadn't had in the past, where all these is the skip spawning piece, and the fact that such a large fraction of the population isn't reproducing every year. It's really hard though to figure out how concerned we should be about that, because we don't have any knowledge of what the historical spawning rates are. So if it's a new thing and all of a sudden half of the fish aren't reproducing, then that's probably kind of important. But if it's just sort of all along been part of their sort of strategy to manage energy and survival and production, then it's not really as concerning. So it's hard to know. It's just another complication in their already complicated life history. But it does seem like keeping track of the age and size structure of the population is important. So to conclude, there are potential differences in some of these rates of these phenomenon during different reproductive years. Again, these old and large female fish may be an important sort of reservoir of energy in the population. The usual science statement where more money, more research, et cetera, et cetera, et cetera. And really right now, it's not really clear how this may or may not impact our future management decisions for the species. I would like to thank the following individuals and organizations for all of their help in this project. And thank you.
Now the hard part of the question is, so do we have questions? I got a question. So one of this year they're talking about reducing the survey for bottom trawl from 3 vessels to 2 vessels. Is actually happening. And so it sounds as if some of the deeper stratum are going to be removed. How do you, I mean, in terms of your species, are you concerned whether or not you're going to have the data to actually keep track?
Absolutely, that's a huge concern for these deep water species, because we're kind of not doing as good of a job as we should be anyways, really sampling the deep water areas. And particularly, yeah, I think that's potentially a huge problem. I know the short riggers, I think there's been some movement to use some of the longline data from the sablefish survey, that might actually overlap some of their distribution better. So I'm guessing that that's probably the direction we'll go in, but yeah, it's not great.
And then, so one other question, 2016 was the warmest year ever compared to 2010, which was on the colder side. Do you think that would affect your results?
Yeah, definitely. I think that environmental factors probably play a big role in whether or not how many spawners there are. And it's interesting because they actually seem to do a little bit better during the warm years, so it'll be interesting to see.
wondering, because I don't know that much about rockfish, but are there, other rockfish species that we have been studied, like baby crabs in eastern Gulf, that have this same skip molting type of process, or, you know, is there any info that this is a characteristic of rockfish in particular, or?
No, this is really, for the deep water species, this is really the first time we've documented it in a rockfish, at least in the Pacific, so. It's something that happens that there's been a lot of research on gadids on the Atlantic side, but not a lot of research on rockfish on this side. And I suspect these rates are gonna be highest in these deep water species. If we see it in the more shallow water species, maybe we're not gonna see quite the same rates.
Okay, one more question.
So, the age difference, I know we don't have a good idea of the age, but so the age at first reproduction versus age at that later stage where they're more successful, what, do you have an idea of what the difference is there?
I don't. That size at which they become successful, 75 centimeters, is really large. So that really is just the really big fish in the population. It's about 25 centimeters bigger than their size of maturity. So,
Okay.
And the size of maturity is probably 20-ish, I would guess. So, yeah, it's probably about a fuller.
Thank you. Okay, so we're moving on to the third presentation. Zoo Archaeological Analyst of Central Alaska Fish Fauna by Holly McKinney. That's a mouthful.
Hi, my name is Holly McKinney, and I'm an archaeologist at the University of Alaska Fairbanks. And I want to talk to you about the project I've been working on for the past year. This is part of an interdisciplinary project, and I'm doing the zooarchaeological analysis of the fish bones from interior Alaska. And it's with the aim of understanding human prehistoric diet over the last 13,000 years in Central Alaska. There are other people on the project who are doing stable isotope analysis on bone collagen, and we're also employing ancient DNA methods. Okay, so I want to talk about this. It's a museum project, so we're looking at curated archaeological assemblages. So these have been excavated before, and they're found in museum collections. So I found, pouring through the literature, that there are about 25 collections that have fish bones recorded. But I'd like to say there are thousands of archaeological sites that are curated from this area, so why are we not seeing fish bones? Well, it's because fish are small, fragile, and they tend to fall through the screen, the mesh sizes, that is the industry standard, so 1/4 inch mesh. So we just don't see a lot of fish bones. And so there's also other issues with like taphonomic preservation, they degrade faster. And so I was able to find 25 different sites that had fish bones, so that's pretty cool. Okay, so I want to talk about model predictions. These are models that look at long-term interactions between humans and fishes in Central Alaska. And then I want to talk about preservation and fish-specific taphonomic considerations. And we will present a map of the sites that have fish bones. And then I'm going to go into some fish bone statistics, and I'm going to keep it light. And so here is the model. Okay, this is something that I developed with Ben Potter, and we divided it into 3 different temporal time periods. We've got the late Pleistocene, early Holocene, which spans from 13,000 to 6,000 years ago. We've got the middle Holocene, which goes from 6,000 to 1,000 years ago, and then the late Holocene, which spans the last 1,000 years. Okay, so during the late Pleistocene, early Holocene, we expect to see a broad diet breadth, so they're going after lots of different taxa. Highly mobile, going across the landscape. And then they're focusing on high-ranked ungulates, which include like bison and elk. And they're emphasizing the lowland areas because that's where the large ungulates are living. And we're not expecting to see storage. Okay, but during the middle Holocene, we expect to see the diet breadth narrowing, seasonal mobility focusing on caribou. So they're going into the upland areas where you can find the caribou. And we're not expecting to see evidence for storage for fishes, but we will see evidence for storage for caribou. Okay, so then during the last 1,000 years, during the late Holocene, we're seeing a similar narrow diet breadth and reduced mobility, but they're focusing on fish. There's fish camps along the rivers. They're still going after caribou, but not as much. And we expect to see evidence for storage for fishes. Okay, so we have to consider taphonomic preservation as well, because fish bones degrade once they're deposited into archaeological sediments. So during the late Pleistocene, we expect to see better preservation. And that seems counterintuitive because they're so old. But they're deeply buried and really rapid sedimentation. So less is blowing in and they're buried and they're buffered from the acidic soil layers. So we get good preservation during that time period. But the middle Holocene, we expect to see poor preservation because there's lower sedimentation rates and we see the influx of forest soils, which are highly acidic, and they degrade the mineral portion of the bones as the bones just disappear. And it's also more highly affected by cryoturbation, which is the freeze-thaw cycle. So bones get moved around and broken as well. Okay, but during the late Holocene, we expect to see better preservation again, largely because it was just a shorter time period. And so the bones haven't had time to degrade yet. Okay, so we need to talk about fish bone-specific taphonomic considerations. So there are intertaxa and interskeletal element differences in preservation potential. So fish bones tend to have a lower bone volume density, which makes them less able to withstand like the diagenic agents that just destroy the bones. They have thin, fragile shapes, so they're more easily broken. Their size is small, they fall through the screen, so they're just not collected. And then they have a lower protein content, but higher lipid content. All these things together make fish bones just less able to be preserved as compared to mammal or bird bones. Okay, so I want to look at some of the sites that we collected the fish bones from, and they were from the Kuskokwim, the Tanana, and the Copper River drainages, all in interior Alaska. Excuse me. Okay, and so when I went through the collections, these are the taxa that I was able to identify. Burbot, salmon, northern pike, whitefish, small salmonidae, that was largely rainbow trout, and then a small forage fish that was tentatively identified as lake shark. So no matter where I was going in any of those river drainages, we found these taxa. Okay, so I want to talk about fish through time. So the blue one is the late Pleistocene, early Holocene, and we have mostly burbot and salmon. But what you can see here during the middle Holocene, no fish whatsoever in any of the collections. So again, this might be because they're focusing on caribou during that time period, but it also could be a taphonomic preservation issue. And then we see a larger jump during the late Holocene, the last 1,000 years of more fish bones. Okay, and so this is how it plays out here. During the late Pleistocene, early Holocene, we see a mixture of salmon and burbot, and a few other of the northern pike and whitefish. But then during the late Holocene, we see basic third split of burbot, salmon, and we see a lot of northern pike. Okay, so I want to talk about fish NISP, which is the number of identified specimens. Okay, and that's just a basic count of fish bones. And from this early Holocene, we can see that they only come from the Tanana watershed, and that is because largely they're coming from the Mead site and the Upward Sun River site. And I was a part of that project, and I insisted that we use 1/8 inch mesh instead of 1/4 inch mesh, and lo and behold, fish. Okay, so during the Mead, or at the Mead site, we have burbot and salmon at Upward Sun River. And so we have a total of like only 480 fish bones for, that whole 6,000 years. Okay, but when we look at the late Holocene bone counts, I know there's a lot going on here, but in the copper, we have a focus on salmon. In the Kuskokwim River at Kodiak, that's mouthful, we've got burbot. And then at the Tanana Valley, we've got Healy Lake with the dominance of northern pike. And we can see the numbers jump up. We've got, 2,500 fish bones. So, it's increasing. Okay, so this is the minimum number of individuals. So you take all of those bone counts and then you say, well, let's put this into manageable units, and these are how many fish we have. So really, we're not looking at too many fish for that whole early period. We've only got 17 fish, and 7 of them are burbot from the meat site. Okay, and so when you look at the late Holocene minimum number of individuals, we've got 4 salmon from the Raylan site, 18 burbot from Kodiak. But the real story is the northern pike. There's 82 northern pikes. Okay, so I'm going through the assemblage, and I see this giant bag, and it's just filled with nothing but pike heads and duck heads. So somebody was out hunting and fishing at the same time and just, dumped it all into one spot. But it was kind of cool to see that. And so our, our minimum number of individuals level just, jumped up pretty high. Okay, so that was more of a gross analysis. And then I was able to do more detailed analysis at the Upward Sun River site, because this is something that I was able to control. And so at this site, we found cremated remains of a 3-year-old infant, and then below that, a double infant burial. And so associated with that, we found a bunch of different cars from the same time period. And so I did a more detailed analysis and looked at all of the bone remains in the hearth. And so I picked out all the tiny little fragments. And here's an example of some of the salmon bones that I pulled from there. And based on the fenestration patterns and the little teeth there, I was not only able to say that they were fish, but they were salmonidae. And when I looked at the individual hearths, the data from there, we can see our NISP number jumps up to 6,778. And our M&I looks, went to 10. But when I looked at the site as a whole, my M&I values were only 1. So what this pattern tells me is that the people at the site were burning the fish remains to dispose of them. Provides fuel, lots of bears in the area, so it just makes sense. So we know that fish were really important to the inhabitants, and it was likely like that at a lot of the different sites. So we don't have evidence. Okay. And so this is some of the papers that my colleagues and I have been working on over the years. And we've done some more intense analysis, and we've determined through ancient DNA analysis that the salmon at the Upward Sun River site were chum, that stable isotope analysis revealed that they were anadromous, and it is near the modern limit of chum distribution. And we were able to determine through sediment analysis, stable isotope analysis and fatty acid profiles of the sediments, that there was recurrent use of salmon in the terminal Pleistocene. And we were able to see it back to 11,800 years ago that they were using salmon there, even though we only have zooarchaeological faunal analysis, or evidence up to 11,500 years ago. Okay, so I wanted to look at the importance of salmon compared to the other fish species, and I developed the salmon index, and a value of 1 means complete total dependence on salmon, and 0 means nothing. They weren't using it. And so during the late Pleistocene, early Holocene, it was under 0.6. We have no samples for the middle Holocenes, we don't know. And it was like 0.3 something for the late Holocene. So what we can say is, based on these curated archaeological samples, that there's no evidence for intensive intensification use of salmon. So, I'll get back to that. In conclusion, what I can say is a wide range of freshwater anadromous fish were exploited in central Alaska during both of the time periods. And so consistent with previous research, we see a greater dependence on fishes over time, so it does fit our model. And as the taphonomic model predicts, fish are absent from the middle Holocene assemblages. Again, this could be, preservation issue. And the late Pleistocene, early Holocene assemblages are dominated by burbot and salmon, and the late Holocene assemblages are dominated by burbot, salmon, and northern pike. And again, there's no archaeological evidence from the curated archaeological collections for resource intensification focused on salmon during any period. But here's the key. If we want to better understand the prehistoric interactions among humans and fishes, we need to focus our excavations along the streams and the lakes. We need to go to the places where they were, fishing. And so this was the first step, was to look at, what's curated, what's in our archaeological assemblages. And so now we need to go back, we need to focus on those areas where the fish are. And we also need to use small mesh screen sizes and be really careful with that and look in the bars and, just be really detailed. Okay, thank you.
So, does anybody have questions for Holly? Right here.
Yeah, do you have any ideas on the number? Were the fish more common early on, or I always think there was less fish back then too.
Yeah, there probably was. One of the main issues is that a lot of archaeologists don't see the beauty of fish, and they didn't collect them. They didn't, use methods that were aimed at collecting them. So I would say that a lot of fish have been overlooked. However, the preservation is an issue. All kinds of things are going on. So it's hard to say. I mean, 13,000 years ago is a long time. So.
But are they coring lakes? You talked about
Oh yeah.
To get the absolute, look at the nitrogen and see if there were how many fish were in the lake relative to today.
Oh yeah, and that is part of our larger project. Bruce Finney is doing lake core analysis from interior lakes. So yeah, we're definitely going to get that information. It's just going to happen this summer. So we can establish that, how abundant fish were in the system. But that doesn't correlate necessarily to, what humans were doing. I think in the past people have really thought that people focused on the large ungulates, the big sexy beasts. But we're finding out that fish were really important in the prehistoric record too, like the late Pleistocene record.
Do you mind going back to your map really briefly? Okay, right there.
Oh, that one?
Right there, yeah. I just, what watersheds, I can't really tell with the possible ancient course.
Mm-hmm.
And it looks like it's going over exposed Beringia. But what are where's the mouth? Is that near Dillingham or what?
That is sort of
Okay.
in
Cool.
there.
I guess one question in my mind is when I look at this, based on current time frame, the abundant fish are, in terms of users in the present time, it's Chinook for the Yukon, and then in the Copper River area, I would think it would be Chinook and sockeye. So to see the chum jumps out is kind of odd in my mind. So I don't know if you have a comment about that.
Yeah, so in the interior sites, they focus on chum and during the late summer or early fall runs, and it's pretty consistent with all the assemblages that, we're confirming it with ancient DNA analysis, but based on the morphometrics, I'm seeing chum.
Okay, thank you very much, Holly. Okay, so we're moving on to experimentally wrangling with fish parasites, and that's Brian Himmelblum from Kodiak here. Go, Brian.
All right, thank you, Julie. Thank the organizers for providing an opportunity to talk about a project we just got involved with last year. I'm a microbiologist, so I work with cultivating very small microorganisms. In this case, we're working with a little bit larger organism. So just to give you a little bit of background, there is a concern regarding fish parasites. We saw an article earlier this year that caused a little kerfuffle regarding tapeworm in Alaska salmon. So what we were involved with was to focus on nematodes. And just to give you some more information, we know that there is an association between the parasite, in this case the nematodes, and the larger host organism. And what this leads to essentially is that there could be some seafood-borne illnesses if a consumer does consume a live parasite. The primary cause is due to consumption of the raw product or undercooked products in which the surviving parasite may be picked up by the host. So in the past, the FDA has provided some guidance in terms of how to control parasites, and primarily it's due to freezing at various temperatures and for different time factors. And the Alaska Seafood Marketing Institute was interested in addressing these previous conditions to see if we could find a way to lessen the time in freezing. So the objective was to monitor fish fillets which were undergoing freezing in our glass freezer that we have at the Kodiak Seafood Marine Science Center. We also were looking at doing some undercooking studies regarding fillets that had parasites that were added, and try to determine when they would be inactivated. So some of the general methods we were looking at, whole sockeye. We obtained whole sockeye salmon and Pacific cod from processors. We tried to determine the number of nematodes and or other parasites in the fish. We would collect parasites and use those by adding those to the fillets that we were working with. We could monitor the internal temperature by a device that will record temperatures over a period of time. We could also monitor the temperature of the griddle that we were cooking fish using an infrared thermometer. And then we would evaluate the nematodes for post-treatment to see if they would survive. This gives you an idea of the eye button that we inserted into the cut fillet. Then we would close up the fish and put those onto our freezing trays and then put them into a glass freezer. And this gives you an idea what the eye button temperature profile would be. We started initially with ambient temperature, and in roughly about 3 hours, it would bring the temperature down to about minus 22 Celsius and would hold that for a long period of time until we would end the temperature recording and download the data. So when we look at parasites in salmon for a small collection of fish that we collected from the process, we can find that there'd be a variety of levels for parasites. Many times we would find absence of parasites. And associated with the fish, we would find primarily they'd be found in the gut cavity, which shouldn't be a problem for processors who are making fillet materials. But again, this wasn't enough for us to collect to inoculate into the fillet material. We did a second survey of fish, and again, we could find very little amounts of parasites to work with. This gives you an idea of what we were looking at in the internal gut cavity, where there would be some parasites shown here. But what we're really looking for are some of the parasites. They're very hard to find. They're associated with the the fillet. Unfortunately, it was the end of the season, so we didn't have enough fish to work with for doing the inoculated study, so we turned our attention to cod. It's very similar. We could find very few parasites associated with the commercial cod species. So that required us to actually go to the processor and have parasites collected on the line in the discard. And then we use those to do the inoculated study. Again, we use the I button for monitoring temperature, and we would place the live parasite in the same area. We'd cover it over, make it kind of a sandwich to make a thicker portion of the fish. Here's some dimensions of what those fish look like. We froze those fish anywhere from 1 to 96 hours, and we used paired fillets randomly placed on the trays, and none of these samples showed survival of parasites, which is a much shorter period of time than the recommendation from FDA. Again, another view of the temperature profile. This is about a 2-hour freeze cycle for the particular fish, and then upon stopping the freezing process, we would thaw the fish out and then recover the I button, download the data, and also evaluate the parasite that we had added. For doing the cook temperature, we also use the I buttons for monitoring. It's a different type of I button, which would work at the higher temperature range. We used our griddle cook, and we would monitor the griddle temperature with a handheld infrared thermometer. And in this study, we were looking at very short cooks, anywhere from a half minute to about 3 minutes. There'd be a variation in the surface temperature. Here are the sizes of the fillets and their thickness. And what we could see after cooking the fish, we would put the fillets into a ice water bath, chill down the temperature quickly, recover the parasites, see if it was live. In these cases where there was very short temperatures, there was survival. The appearance of the fish, again due to undercooking, would show that the fish were indeed internally appeared to be raw, until we would get the temperature a little bit higher, a longer period of time, that showed that the parasite was not as active as the previous ones in the different portions of the fish. This gives you the example of the temperature monitoring going from ambient, 3-minute cook, and then ice water chilling of the temperature. And this is what the product looked like for that 3-minute cook. It apparently looked visually as a cooked product. Here's the parasite that we added along with the I button. So in conclusion, what we found out is that the freezing method does work, but it is much quicker than what the FDA has indicated as proper freezing to inactivate parasites. And we also showed that the undercooking could be a potential risk if live parasites are found associated with non-frozen fish. So some future needs, it'd be interesting to look at thicker portions of fish because it takes a longer period of time for the freezing process to occur to freeze fish, maybe even thicker packed fillets. We'd like to conduct a heat activation, again, to show which temperature these parasites are inactivated. And it would be helpful to have some type of molecular identification of these organisms for definitively identifying the parasites. So project funding came from ASME. I want to thank Michael Kodiak for insight into this project, and of course the anonymous seafood processor for providing the parasites. Thank you.
So questions for Brian? Okay. Go ahead, Julie.
I find this topic
interesting. So what are our typical freezer, the normal person's freezer temperatures at home? I mean, we're not reaching those last freezer temperatures for the average person. That's
Correct. It may be anywhere from -5 to -10 Celsius. So yes, the home freezers are not necessarily capable. It depends on the manufacturer of the particular freezer. But in the home, those products are usually kept frozen for a longer period of time, so maybe the time temperature factor works in favor of the consumer for having inactivated the particular parasite.
And also, did I understand that you had all the parasites destroyed in the cod, the parasites that were inserted into the cod fillet, they were destroyed in the freezing, even with minimal freezing?
For minimal freezing times, yes.
Do you think that there was any artifact of opening the fillet and removing that parasite in and out and already handling it prior to?
That's a great question. I mean, the physiology of taking a nematode out of its natural resource and holding it and then transferring it. Earlier in the summer, we did some holding studies by holding the obtained parasites in a phosphate buffered saline at 4 degrees Celsius, and we could maintain those with changes of the buffer solution over a period of weeks. So it's possible that the organism would retain viability in our operation. It's just a matter of having that freezing temperature that inactivated the particular worm.
So I'm going to ask the question. So when you say that there's a risk, that the parasite is still alive and you ate that fish, what is that risk?
I don't have data to show what would happen in a particular specific case, but it's hard to know. It depends on the health status of the individual. It also depends on the species of parasite that is being consumed. So it's hard to document who would be affected by these particular parasites in any one case. But we do know from the literature that consumption of raw products, particularly in other countries, allows a high incidence of parasites to be consumed, and a number of people come down with foodborne illnesses from this ingestion of the parasite. Or parasites, it could be multiple that are being consumed.
Any other questions?
I'm curious, what's the next step? Is this information to be provided to the industry or to the FDA, or is it called for a follow-up larger study?
It could use, yes, it could use a future study to build on this particular initiative that we had started. The information would be provided for ASME to provide to the industry as guidance and for feedback also in terms of the kinds of issues that they're facing.
Okay. Anything else? I guess it's time for the break. And what time do we restart?
Come back at 10:45.
Hi folks, we're going to take a break for the morning, our mid-morning break. Let's come back by 10:45. I think we ended a tad early. It's a great opportunity. You can fill out that survey or keep that in mind as you go out and visit with your friends. It'd be great. Thanks.
Yes, or we can put a number on it. See what number you are. It can't help. Yeah.
Check again, we got it off of the, sure. We know it works this way. Yeah. Who knows what that was? That was perfect. Final slide. Good, great.
so busy and important.
Well, it's nice to see.
Yeah, I was in another meeting in another building.
It's sort of like exercise. I don't feel like good like afterwards. This guy's like, it's a boom. Exactly. So now we're working on effort to give them all their own separate little containers. It's a lot of we're using little Tupperware containers and well, I don't know. 300 yesterday, so today. We've got so it's just, we got a PVC pipe and then we've got zip tied all these Tupperware containers stacked up. So you've seen our tanks downstairs, so they're just upright in the air. So because you got to be able to pull them up and check for molting. Oh yeah, yeah. Well, they're, because we want to hold them, so, because no, they're all known, and we know the size of the pad that went in, and then once it molds, we know the size. It's that self-giving. I think so. Hopefully are going to be done today. No, not today. It's just hard to predict. They're going to run out of something. Well, I think today the usually I emails first thing in the morning, have a cup of coffee. And this gal, Christy, is came from she's oh yeah. Yeah. And we just the faster. So did you say you talked to her? Oh yeah, because he since they brought them. This has been kind of our she's not a good crowd live. Very seen here. And they always have coolers and they sit in some Anchorage and they don't have transfer or stuff. So this was great for Sunset Bay. Brought it here, they're all inside, brought it here. We've had very few I mean, on arrival. I see some of the ones that are put things yesterday. So they were sleeping on their backs.
Hey Lori, what's up? I'm good, how are you? And the answer
here?
is no, I'm sorry.
Yes,
So,
although I had one of my, my EFH maps was shown in, Matt's presentation, so, I actually did have something.
I'm sorry. Take that, make it right. I dropped that ball. Oh, let me see. Let me see if I can find the numbers, and then I will search, Karen, what is Karen Stevens?
There's supposed to be somebody on the surface. I know that before when I was at Wood Diner, we didn't have to have somebody fully suited up. I know the rules have changed there, and it might be, and I don't know what it is. I no longer know what's required. I don't have an AA event certified. The thing is, I'm on the other side of the shop, right? So I'm not here, I'm not in the box, so it's not a priority. I agree with mine. I'd like to help out, plus I'd like to be able
Yeah, and that's
so that if I'm not with running,
Maybe rock kind of
That was the answer.
You don't got a complete thing. How about you?
Oh, no, maybe two sessions. So you're this year,
Oh, I see.
Sorry. No, I'm working on a lesson plan. I'm putting together for some teachers. No, I'm just shooting it out there. This computer is going to all schools in the district in this week. Yeah. I mean, it's actually live streaming.
a lot of things.
And meanwhile, you're right now, everything's happening. Yeah. Today was kind of one of those.
Are you putting this, are you going, am I going to? Is this going to all the schools?
This one is, and this one is live streaming to YouTube.
I think that's.
Oh, wow, so are we in classrooms a lot?
It's actually right now only 3 rows. And
I already did that.
Kodiak classrooms are around the room. Larson Bay and I think the high school.
Yeah, I did it, I gave it for Well, no, they were all There were kids, I felt bad, but I didn't, there's one problem with that whole thing. I didn't know what the kids were from at first, but then I started talking about what, Larson Bay and Larson Bay kids.
Oh, right. They like to hear about
I know, right? Teaching people how to wine for coal and I mean, some where it's like from that stuff happened,
Hey, everyone.
Yeah, exactly.
Hi, if everyone can take their seats, that'd be really helpful. Get back on track and keep moving forward this morning. So please grab your coffee.
Yeah.
Julie Lonnie will be continuing as session chair.
Probably sounds fun. Yeah. I keep you guys talking to my college.
Okay, moving on session 3, dynamic methods. And so our next presenter is Mary Beth Lowen with Alaska Department of Fish and Game, and she is the sole Alaska Fish and Game presenter, I believe.
No.
Oh, I got that wrong. So anyway, thank you very much for being here and making your presentation on Chignik River Post Weir enumeration with Ditson.
Thanks for having us. Yeah, Callie Spellinger will be talking tomorrow, I believe.
Yeah, and April.
So, for okay. All right. So, this is the Kodiak Area Marine Science Symposium, but this work actually is research that we've done out in Chignik on the Alaska Peninsula, down here to our southwest. Chignik is a large watershed. It's got the Black Lake at the head of it, runs down into Chignik Lake. There's a village here at Chignik Lake. Through Chignik River, there's a village here in the lagoon. There's 2 large commercial fishing areas, Chignik Lagoon and then Chignik Bay out here. So this is where the focus of this talk takes place. Many of you probably know that Chignik is a seine fishery. It's the Chignik Management Area. There are 2 runs of sockeye salmon, an early run and a late run, and there's also local pink and chum systems. There's about 96 streams that produce salmon, but sockeye is the main commercial and subsistence resource in the area. The early run and the late run both are fairly large. Their 10-year average is over 1 million fish each. And again, it's a very important subsistence resource for local area residents. The Chignik management area is managed on site through the use of this very large unconventional weir. It's over 400 feet long, spans the Chignik River in a tidal area. It's put in every year and removed with a pile driver. We sample fish through the use of an in-weir trap, and if I can find my arrow. There are 2 cameras installed in this weir, and the fish are counted the first 10 minutes of each hour by species as they pass through these gates. So the gates are always open. It's unrestricted movement upstream. But it is limited seasonally. As you can imagine, keeping this huge facility, this huge structure in river takes a tremendous amount of manpower. It takes a tremendous amount of funds, both to put it in and remove it. Late season, there is a buildup of spawned out fish, which build up on the weir. Also requires a lot of time and manpower to keep the weir clean. In the fall, typically there are large rains, which can cause high water events, which also means that the weir is at risk of being washed out. And so after the weir is removed, typically in late August, due to restriction of funds and, as I said, all of these other environmental factors, we have used a simple statistical model to estimate the fish that will be escaping upriver after weir removal. And what that is, it's a time series model, and it uses the rate of decay in the last period of time that the weir was in the river to estimate the escapement that we predict would happen after the weir is removed. The problem with this is that this statistical model can only use the information that we have, so the rate of decay and escapement before the weir was removed. And there's anecdotal evidence that there is actually a late sockeye run that hits the system in late September, early October. And there's also no way for us to actually validate whether this statistical model is accurate, right? We just have to take this as our best available method for estimating the fish that are going to pass upstream in September after the weir is removed. So, in order to validate the statistical model, we have come into using a Ditson in the river. And a Ditson, for those of you who don't know, who haven't heard me preach the wonders of Ditson, it's a sonar, and before you say it's just like an ultrasound, it is just like an ultrasound. It's using sound through water, it hits rocks and logs and fish, and comes back to the transducer as imagery, and we are able to count rocks and logs and fish. The benefits of Ditson, you can see here, it's small. It's about the size of a large shoebox. It weighs about 15 pounds. You can put it in the backpack pretty easily. It's non-invasive, so it's great for places where you have high flow, or you have, you couldn't put in a typical traditional weir because of changing river structures, tidal influence, things like that. It also doesn't care about turbidity, so it's great in areas with glacial flow. And it has a pretty long range. It has a working resolution of about 40 meters, but it does do detection out to 80 meters. So to give you an idea of what this looks like, for those of you who haven't seen it, I'll orient you here. Shore is at the bottom, and offshore, 10 meters here, and you can see these salmon swimming upriver, right? So this is what we look at in real time to estimate fish moving upriver through our sonar. So our objectives for this project, which has been running since 2011, are fourfold. And the first is to estimate the population size and run timing characteristics of sockeye salmon and coho salmon moving upstream after weir removal. And to do this, we have 2 Ditsons, one on each side of the river. You can see here looking across each other. So we do have full river insonification. And like projects throughout the state, we record continuously. So we have full 24-hour files created. But we only count the top 10 minutes of each hour, and then we extrapolate that to get hourly and daily counts. And the Ditson was originally developed by the Navy. They do great work. But now it's used throughout Alaska. Alaska pioneered this for the use of salmon counting, and we have about 28 sites now throughout the state, and it allows us to count all the fish, right? So you can imagine, though, when you see something like this, and you're sitting in your weather port, and you're looking at fish going by, you could try to count these with a little tally marker, but you'd quickly get carpal tunnel. So instead, what happens is we take that moving imagery and we expand it into an echogram, basically, which is a static count. So you can't see it in this screenshot, but there are fish here. Those are translated out into our echogram, and we're able to count these digitally. If you can use your imagination, this is supposed to be a salmon swimming upstream. This is the tail, this is the head, and this is a salmon swimming downstream. Here's the head, here's the tail. So we can get a lot of information. We can get direction of travel, we can get distance offshore, and we can also, through Didson, get some speciation. So this is a quick file, but let's see if I can find it again. This is a school of smolt coming downstream. Maybe I can't show you. There we go. A school of smolt you'll see coming down, and then they move offshore and get a little deeper. The red disappears as they get a little farther away from our sonar face. These are flounders moving upstream with the tide cycle. I'm going to do a little interpretation for you here. I have spent a lot of time looking at sonar, so I can recognize this easily, but you can see that they are moving differently than those salmon files that I showed you. And you can actually hopefully see the pec fins and the tail fins and see that these are different kind of fish. These are not salmon, right? And then this I've put in just for fun. This is a very large salmon. I've actually sped this up, but you'll see we get seals that come and play with our sonar. They really love checking out what's in the water. So we've got this guy here checking out our sonar face. And what I love about this clip is you can see the resolution. You'll see his tail flippers here pretty quickly. And he's just hanging out, swimming around. So we can look at these species and see some of it. But when it comes to salmon, in Chignik, we don't have a size difference between our sockeye and our coho, and we also, it's a small river, so we don't have area of river travel to segregate these fish. In the Kenai, they can see king salmon and sockeye salmon segregating by distance offshore, but we don't have that in Chignik. So how do we tell what kind of fish is it? In Chignik, we are actually physically sampling for fish. We use a variable mesh gill net here, and we started this project fishing at several sites throughout the lagoon, but we've condensed it down and found that our best fishing spot is here at a place called the Jaws, which is quite near the weir. We head out and we sample across a high tide. We also have a beach shade, and we also rely on subsistence reporting to kind of validate that what we're catching on a daily basis is lining up with what the local residents are catching. And from these catches, we are able to collect age, sex, and length, which helps us build much better brood tables, because when the weir is pulled out, we lose our ability to sample these fish. So our late season sockeye, we don't have the age classes and the sex ratios, right? And we also don't have that information for coho pretty much at all. So you can see here over the years, we've been able to sample several hundred fish. We've gotten better each year. 2016, this is just the 10 days after the weir was removed. So we got almost 200 fish in 10 days, 200 sockeye, excuse me. So we've been able to really add to our database for our brood tables. Which brings me to, you know, we can see all these fish. Is it better than what we've had? And I'm showing you, this is 2015. I'm going to talk you through this. This is the weir counts here. The weir was removed on August 23rd of 2015. We had some funding restrictions. This is our statistical model with this nice, smooth rate of decay, constant rate of decay based on the decay in escaping counts from the weir. These are our Didson counts, right? So when you look at this period from September 1st through September 15th, you can see a real difference between the statistical model and the actual Didson counts. Because Didson is looking, it's a real life count, it can incorporate these daily fluctuations in escapement magnitude. And what we see is that in 2015, the statistical model would have estimated just over 15,000 fish, but the Didson for the same 2-week period counted over 132,000 fish. Right? So that's a big difference between the two models. And then after the September 15th, we had almost 50,000 more sockeye come through. So that leads us to, that makes it seem like, wow, we should be doing this all the time. And a comment I have there is, in a year when you have great funding and normal run timing and a normal escapement magnitude, it may not necessarily be prudent to supplement your escapement monitoring with the Didson. In 2016, we had this great combination of extended funding, nice weather, regular run timing, The weir came out on September 13th, and you can see that before that time, we'd already had almost 335,000 late run sockeye. The additional Didson counts for the next 2 weeks were about 17,000 fish. So not a huge proportion of the run coming in after the weir was removed. Same for coho salmon. The coho run was about halfway done by the time the weir came out. But in a year when you don't have very good funding, so you have to pull your weir early, or you have extremely late run timing, or as in 2015 when we had the combination of both, the Didson becomes extremely valuable. So here we had to pull the weir, and I say we, it wasn't me, it was the management crew. The weir was removed August 23rd. Up to that point, 308,000 sockeye salmon had passed. After that, 303,000 more sockeye salmon passed, almost the same amount, right? Coho salmon, the coho run had barely begun. Less than 3,000 had passed the weir. After the weir was removed, over 60,000 passed the weir. So in 2015, it was really important for us to be there and see these late season fish. It was a really late season and a lot of fish coming in. So do we need to do this? And I would argue, yeah. In years when you have normal run timing and great funding, you know, so that you're not going to count that many more. That's fine. In years when you have small aberrations from your normal run timing and normal escapement magnitude, you know, it's significant, but it's not going, you know, if you have a run of over 1 million fish, 15,000 additional fish or 15,000 less fish, not such a big deal. But when you get that nice combination of reduced funding and strange run timing, which as we know is happening more and more, it can be really valuable, right? The difference between our statistical model and our actual counts was over 200,000 sockeye salmon in 2015. So looking forward, the Alaska Sustainable Salmon Fund has granted us funding for 2016, 17, and 18 to keep the weir in later into September, through mid-September, so that we can get some paired Didson and weir counts for calibration factors. And the hope there is that we'll be able to do comparison of weir counts and comparison of Didson counts, develop some, some calibration factors, and then in the future we might be able to just use the Didson for a much more extended period of time, which would reduce the funds required and reduce the manpower required to keep that weir in all season. There's also some implication or potential implementation in other rivers around the Kodiak area. We've used it in the Ayakulik when the weir has blown out. That's a really flashy river system. We've put it over on the North Peninsula in areas that are really turbid and we can't see from aerial surveys, and it's a great little method in, as I said, glacially segmented rivers. It's also been, we've documented some smolt trap avoidance. We've looked in the Karlik for smolt enumeration actually, and Didson doesn't require very much manpower. It's very low cost to run. We actually have 3 units here in Kodiak that can be used around the area. But as you all know, Alaska is kind of in a fiscal crisis, and so putting this into new projects is probably not in the cards for us right now, simply because we don't necessarily have the funding to be going out and doing all these amazing new projects. So that pretty much encapsulates our work in Shignick over the last several years, and they have 2 more years of this project to go thanks to the Alaska Sustainable Salmon Fund. And I'm just going to leave you here with this little clip, and then gratefully thank the management staff and Luke Hensley, who will be running the Didson this year. Don Wilburn and Lucas Stump are in charge of keeping that weir in and managing the Shignick management area. Here in Kodiak, Birch Foster and Kevin Shaberg are involved in this research, and of course our fabulous seasonal staff every year. That's all I have. If you have any questions, do I have time?
You do.
Oh, all right.
I want to note that you have 20 seconds left for the top part. So is there any questions for Mary Beth?
Because I can talk about sonar forever.
Over here, Gordon.
Yeah, very nice. I'm just curious, are there many salmon beyond the 10-meter range of the Didson? And obviously they can go up or down, and how do you deal with those? Or is that the comparison with the weir that you're going to do?
That's a great question. So in Shignick, we have been able to reduce our counting window to 10 meters because we actually have looked across the whole river, figured out that on each bank, on one bank, 93% of the fish are within 10 meters. On the other bank, it's 95%. So we reduced that counting window down because the resolution is so much better. As far as upstream, downstream migration, that is definitely a problem. Because this is non-invasive, of course, the fish can move move with the tides, and we've actually put this in at the mouth of Karlik during when the coho were starting to come in, and you just see these big whirlpools of fish going up and coming down and going up and coming down, and that gets to be really time consuming. They do have now, the next version of the Didson, the next generation is called Eris, and it has some pretty great auto counting with direction, so that's supposed to help with that requirement, but it does. Once you have fish moving up and down, the amount of time required to count is just exponential.
So is the software tracking the fish then once it sees the fish, it keeps track of that fish through the counting process?
In the new generation, the Eris, yes, that software does. In Didson, we don't have that software capability. So if a fish comes in and mills in the window, it's the responsibility of the actual technician or person who's counting to keep track of that fish until it crosses out of the beam and then mark it as upstream or downstream.
So the little icons on the image, those were pointed and clicked into
Exactly.
place?
Yep.
And not recognized sign by the machine?
Yep. With the Didson, it's all manual. You can do an automated counting, but it will just give you a count of image or count of traces basically without upstream, downstream. And of course for us, we need to know how many are actually going up to the spawning grounds. Does that answer your question?
There's another question.
Similar to that, what's your processing time on the from looking at it real time to then counting average?
Yeah, I will say that you get pretty quick with experience. Yeah. And of course it all depends on how many fish are in your file. It also has a capability where you can subtract out all the blank space basically, so you only get movement within your file. It's hard for me to quantify the amount of time it would take, but I know with the crew that we had on this last season, they were all pretty quick, and we could get through both banks about 75% of a day within that day. So the ultimate goal would be for us to be able to provide real-time information for the managers. We're not quite there yet. Yeah.
So I want to ask a question. So as you look forward, you see Weir's going away and Didson's being the technology for counting. I mean, what's the cost basis one versus the other? Is one 10 times more?
I don't have those numbers. For Kodiak, our weir systems are, they run smoothly and they're great ways to get information. In the rest of the state, I do think that people are going more towards this kind of technology. You do still have to have, unless you have a perfectly clean system with only one species, you do still have to have a way to get species apportionment, so you need to have people on hand to either physically handle the fish or to see the fish. And then of course to get biological samples for our brood tables and age sex length. We'll see how, and now there's drone technology too, so if you could do a drone for speciation and sonar for enumeration,
who knows?
The future is wide open. Well, thank you very much.
Thank you. The next presenter is Patrick Saltonstall, hunting seals with nests 5,000 years ago.
Yeah, and this is basically some research that the Aleutic Museum's done through our community archaeology program. We've been digging at the site right here for the last 4 years up near Sloaney Creek. And I'm only going to present like a really focused part of what we've learned there. But I think it's the most interesting part. But first I'm going to get to that, but because a lot of people, don't know who the Aleutic are maybe, there's a lot of different, I'm going to do a little bit of an introduction about the Aleutic and what I was trying to learn. And so here's my Aleutic in a nutshell. In fact, I encourage you to come to the museum at lunchtime, and I can maybe give you a little bit more of a tour on this stuff. But basically, as on Kodiak, we break up the prehistory into 3 general time periods, and I like to think of them as ways of life. 7,000 to 4,000 years ago, and that's mostly what I'm going to be talking about later about the seal hunting. The Neolithic people are pretty much sea mammal hunters. A band would live in one bay, people would have a seasonal round within that bay, but mostly seal hunting marine mammals. That was the way they made their living. Later in time, from about 4,000 to about 800 years ago, that's when people started to live in big villages, and there was a lot more fishing, so you could sort of think of them as fishing villagers. And then, the last 800 years, last 600 before the Russians arrived, people lived in much bigger villages, and you would have multiple families living in a village, and that's when you start having a lot more, you have chiefs in a village, and you have rank society, and you have a lot more warfare and stuff going on. So that's sort of, but we're going to be focusing on this time period. And it's, Neolithic are really complex, people who live on the land, you have to have pretty complex gear to live on the land. But it's worth emphasizing, what people were using 7,000 years ago and what they're using 200 years ago, it's always changing. And so basically when we're doing archaeology, we're sort of like looking at how people did things at different moments in time. That's part of building up the history. And a lot of that will key into what the climate was doing, what species were present, et cetera, et cetera. So it's sort of an interplay to figure out how it all works. And, not just the gear, but the seasonal round, how the Neolithic lived, how they use the landscape has changed through time. It's constantly changing. This is the seasonal round when the Russians first arrived and they recorded what people were doing. And I always think it's kind of funny. I mean, like, the whole thing starts off with fur seals. Who does that anymore? You couldn't start a seasonal round with fur seals anymore. Although I gather they are offshore still, close enough to hunt. But, so the seasonal round also changes through time. And we're going to be focusing, I think the time is right around in here, the site that we're at. And our community archaeology program is focused on the archaeology for 20 years, the archaeology of one bay. And we've been digging all these various sites in these different parts of the bay. And we've been choosing to not just dig at village sites. We've been trying to dig at all the different types of sites and all the different seasonal sites that people would be using to sort of see how they use the bay through time. And we can also look at how the gear that they use to live on the landscape changes through time. And the last about 8 years, we've really focused up here at the head of Woman's Bay, and that's where the site I'm going to be talking about. And here it is, actually. I took this out of a helicopter when they had the Kodiak went aground. So I was coming back, some good came with that helicopter ride. I got a really good picture of the site. You can see it's way inland. This is the road. Here's Saloni Creek, and the site's way up here. And it's worth emphasizing, when we first did our archaeology at the head of Woman's Bay, I thought we'd just find pink salmon, we'd find a site, we'd find a whole lot of ulus, and we'd say, oh, they fish for pink salmon in the late summer, because that's what you get if you want to fish it. But what we found is we found a ton of different types of sites. Found sites all the way up to the rifle range. Here's the gate to the rifle range out here on Bruin Point. We found a wide variety of sites, and they were all very different. In fact, the only one where fishing took place was here at Bruin Point. That was the site where we found like predicted net sinkers and ulus for fish processing. But all the others were very much confused us, actually. We're like, what are you doing at a site this far from the coast? And we kind of figured, at first I thought maybe the sea level was higher and that the whole valley was an inlet. The shoreline went way up the bay. But what we discovered, but then we did we shot the elevation of the ground in front of the site here, and it was like 12 feet higher than modern sea level. So we're like, oh, sea level was never that much higher. I got an argument with Gary Carver on this. He was right. And then I kind of figured it out. We kind of came to conclusion. I looked out here. I haven't done it. I'm not absolutely positive yet, but I think it was a lake. It was a big lake up there, and there was like a berm. At the head of that field, you see that's all field, and you see the trees. I think the berm is right about there. And so it would be a place like sort of like Rose Teed Lake, with a the short run, and the seals can follow the salmon up into the lake. And it's worth emphasizing, this village here at the rifle range gate was so much more, so we found houses, we found stuff for making, needle type things, and also a variety of tools, because it's a place where people lived. Whereas these sites up on the lake that I'm going to be talking about, we didn't find as much in the way of houses and stuff, and it looks like they were like places where people went to hunt. And I think they were hunting seals, and I'll sort of show that in a little bit. So here, this is the introduction to the Cachavero site, and this is like the wall of one of our, we did a lot of excavation. You can see, these are all our blocks on the, this is the Slowing Creek for scale. And that's about 600 feet that long. And these are all the areas we excavated. You can sort of see that's the wall. And all these different layers are different moments in time. Like we found stuff from, we found a cow actually right under the Katmai ash. So it was like we found a Bruin family cow, and then we found some late prehistoric stuff where there was more fishing. And then the layer I'm going to be talking about is right in here. So this is our level, sort of, this stuff is even older. Down here we had actually a 7,000-year-old house. But this was a period we found all this charcoal and rocks and a lot of bayonets for hunting seals. And this is the time period I'm going to talk about. And I'll tell you, my conclusion is that they were hunting seals with net and smoking them or drying out the meat on site. And then they were probably taking it to their village somewhere else in the bay. This is the main smoke processing feature we found. It's not just like one feature, it's like multiple little pits in one area done constantly over time. And there's a side section of another pit somewhere else on the site. You can sort of see charcoal, then dirt that's burned, and then gravel, and then dirt. And basically with these smoke processing features, you would make a little shelter maybe, a little feature. You'd get a fire and you'd fill rock in, because you don't probably have all that much wood. So to conserve wood, you'd use rock to hold the heat. And then you'd cover it with dirt to sort of dampen the fire, so the fire would reduce the oxygen so the fire would last longer. And so we've kind of gotten to recognize them pretty well. Actually, I was talking with other archaeologists. I don't think they're recognized widely what these things are elsewhere in the state, but we've gotten pretty good at recognizing these smoke processing features. And this whole area, all these rocks, this is all smoke processing going on here. And I should add that we found a lot of sea mammal bones in these features, so we're pretty sure they were smoking sea mammals and not fish. And another thing that I found out from talking to people, I guess in the interior of Alaska, people use some sort of smoke pits like this to make their hides, to sort of, as part of the tanning process, to make them more supple. So that might be going on here too. Here's some of the calcine bone, and here's Justin, who's in the audience, digging up one of our sea mammal bones. You can see bone doesn't normally preserve. And Kodiak soils are really acidic. So the only time you get bone that will preserve is when you have clam shells that reduce the soil acidity, or you burn the bone. You basically are turning it into, you're firing it. We call it calcine bone. And then you can actually recover pieces of bone. This bone here is 5,000 years old. And these are the types of tools we're finding. And so I'm sort of explaining the story. This is how I came to the sea mammal hunting conclusion. And basically, this is the biggest clue. These are the artifacts we're finding. Pretty much that was it. We're finding big old lances, and a lot of them were completely whole, not broken or anything. And we're also finding these, what we call side blades, which are basically the prehistoric version of an ulu. It's sort of a knife for cutting up seals. This is what you call a piece esquier, or a bone wedge, and you use it to put in the end of a bone and split it. You can bang on the other end. And it's like for breaking open bones, so you can use the bone for raw material, or you can eat the marrow. And then we're also finding these utilized flakes or scrapers, and this is for working the hides and cutting and et cetera. And that's pretty much all we found. We didn't find a lot of things that you would find in a house, like needles, or you wouldn't find a needle, but the other types of stuff that you find at other sites. So it really was, they were just coming here to kill seals and cut them up and process them. Oh, where did it go? And it's kind of fun. We found an amazing number of complete ones. And that also argues, if you dig a village site, usually they're all broken. But here they were mostly complete. And I also learned a little bit, these serrations, I was wondering what the heck they were going. They're not for barbs, for holding. And I realized it's probably to decrease suction. So when it goes in there, you can pull it out quickly and promote bleeding. And It's kind of good technology. And also another kind of cool little sideline story. We found a lot of these like non-functional bayonets and knives. And it's like, they're little toys. It's like Junior that was there with Dad learning how to hunt seals, which I thought was kind of a cool part of the story. Then the net part, now this is kind of exciting, but these are probably the oldest nets ever found. Net gear in Kodiak anyway. And normally if I found that at a site on the coast, I think that was a line weight for your fishing rig, for fishing for cod or halibut. But we're finding these so far inland, and we found them all in that smoke processing area. And they're different than nets because they're, net weights are usually on the long axis and they're thin. These are kind of heavy cobbles and they're down here. And also the other thing is you're not going to find nets until the next time period when they're fishing. But I think these were for sea mammal. And we also found this huge one, which didn't really match. And it's like the anchor weight on a net. And I think these were used as the seals chased the fish up the river. They had these nets out and they'd snare them and then they'd stab them with those big spears. And I looked at, I've read in the ethnographic accounts. This is a net. It's at the Kunstkammer in Russia, and it was used for fishing for seals. They generally, in the Russian time period, they'd go on shore and they sort of chase the seals and they'd all run into the water and then they get caught in the net. And I kind of found it cool reading the ethnographic accounts. They like sinew for fishing for seals because it bended and it wouldn't break and snap, whereas they used nettles for fish nets because they were much more sturdy and they didn't require the elastic strength. I had included a very young version of Matt Vendale, so it tells you how long we've been taking. And this is what we didn't find. These are bayonets we found like at the outlet site up by Buskin Lake, and they're using the same spears from the same time period to stab fish. Basically, they're gaffing them in the river. And we also didn't find, this is the layer, that's the lens that represents the 5,000-year-old time period. This is at the outlet site up in the antenna field. And they're not processing these fish in smoke pits. They're basically just catching them haphazardly with their sea mammal hunting gear and then eating them, not really smoking them or anything. And I just included this because this shows you how people gaff fish. This is from Lipnick in the 1950s. And that's how you would get those battered spear tips. And we're not working in a vacuum at the Kashgar site. We found these smoke processing features at a lot of other places. I think Daniel's up there. This is at Old Harbor. We found a 7,200-year-old one. You can sort of see that black lens right there. That's a smoke processing feature. This is across the valley at the Onyx site. This is a big one that had a roof and all these bits. Basically, a lot of these smoke processing features would look like this. You'd have the fire pits in here, and you just kind of keep the meat dry, and it would get smoked as a sort of a side consequence. But anyway, so I'll wrap up. I only have like 30 seconds, I think. What's kind of cool about it is, what I like is like, you always think of smoking meat and stuff. You think of smoked salmon. But, the smoked marine mammal has a far longer history on Kodiak, and that was pretty much what people did in the early days. They didn't smoke fish, they smoked sea mammals. I think we got to try it, actually. I never have. But that's it. And that's the view from the site. We had a kind of nice view.
Okay, so we got any questions?
Yeah.
Okay, so at the other sites that have marine mammal, that you're saying marine mammal smoking areas, are you finding bones or is it interpretation?
It's a lot of it's interpretation, And at the very bottom of the site, we did find salmon bones, although I showed them to you and I don't necessarily agree. But a lot of it is, we have a hard time saying whether it's, definitively. But we seem to only, whenever we find calcine bone, it's always, actually one time we looked at the calcine bone and it turned out to be fox. So, I mean, but it's mostly sea mammal. It looks like it's sea mammal. And the gear, I think the artifacts tell the story more than anything here.
So was it the difference in the bayonets, being pointed versus battered?
Yes, that's a big point, yeah.
Okay.
Yeah.
I had a question related to Holly's presentation. She told us that the fish bones are often missed.
Yep.
So are you seeing that here?
And they might not calcine as well either. they're not as big, so they're way underrepresented. So I would be very skeptical of my just finding sea mammal. I don't think that by itself would be a good enough part of the story. Yep.
What clues do you use to decide a potential site for excavation? Is it like a needle in a haystack, or do you have some remnants at the surface, or some landform, or what is it you're looking for to decide to invest your energies to excavate?
Well, your question, and we were digging a variety of sites because we were interested in how the landscape was used through time. But if you're digging a village, so you got to decide what kind of site you want to dig first, But then to find sites, most of them are pretty, they have different vegetation. Sometimes you see house pits. Another clue is, would you want to use that place today? I mean, most sites are, like the marine debris thing, I noticed that all the sites are located with the marine debris collected. And I was like, oh, that's because people want to live there because they could get driftwood, But this site was actually hard to find because it's not a place you typically use today because the landscape's changed so much. But then I just sort of looked and I thought, if there was a lake here or there was an ocean here, where would I want to go? And I'd want to go someplace flat. And then I just dug holes until I hit something. I got lucky.
Was there any way that you could prove that there was a lake there?
And I want to try that. We tried to do it. We went down there and dug, but the one problem is that river is meandered and it's like eroded everything. And also when we dug down, we hit water table right away. But I did see a lot of lacustrine sediments, like this gray clay in places, which I interpret as like the sediments that would deposit out of the lake, slowly. And actually what I think happened to the lake is that there was an eruption, a volcanic eruption around 3,800 years ago that was equivalent to Katmai ash. And I think all that sediment flowing down Saloni Creek filled the lake up, and I think that was the end of the lake. But I do want to prove it. I don't have it absolutely proved yet. But why would you be hunting seals up a creek that far from the ocean? That's the only way I can think of it if there was a lake out there.
So I guess I have one question. So would they smoke the seals, but do they use it for anything else besides just to eat?
Everything. I think they use the hides for their boats and for clothing, the sinew for making more seal nets, the bone for making harpoons and different tools.
Do they have oil too?
And the oil, yeah. And you can store your fish in oil, or you can store chocolate lily bulbs in the oil too.
So that because of
and the stomach was a big container. You stored stuff in the stomach.
So they have a big more use than like salmon.
Yes. Although the salmon skins, they use that for like clothing and stuff. Yeah.
Okay, so thank you very much, and I guess we'll move on to the next session.
Thank you, everyone. I encourage you to stick around for a couple more talks this morning before we break for lunch. We're going to be moving straight into the community and monitoring discussions and presentations. And I would like to introduce Teresa Peterson. Teresa is with the Alaska Marine Conservation Council and also a member of North Pacific Fisheries Management Council. She's very active in our community with fisheries issues and marine protection. I also want to introduce along with her David Holland. He'll be the first presenter of the morning, but he's also going to be our facilitated discussion leader. So yeah, with that, I'll let Teresa take over, and we'll get a couple talks before lunch. Thanks.
Thank you, Julie. Well, good morning, everybody. It's great to be here for the second full day of the Science Symposium. It's a beautiful day here in Kodiak. It's nice to see everybody out and about, and really grateful for Sea Grant for hosting this Science Symposium in our community. I think it's a really valuable learning opportunity. So we have a great lineup of speakers coming up, and the first up is Davin Holland, and he's from the University of Alaska Fairbanks, and will be providing a presentation, Building Networks to Bridge Information and Action on Alaska's Coast. So welcome, Davin. Thanks for coming to Kodiak.
So Teresa's going to keep us on track this morning and this afternoon, and what I'm going to do is sit over there and try to think of intelligent questions to ask as the discussant when we're all done here. So we've got quite a variety of presentations, but all of them deal in some way with working with communities and tribes and others to kind of bridge those gaps and build collaboratively build knowledge. And so that's what I'm going to talk about today to kind of lead us off, and then hopefully kind of wrap that up this afternoon, 4 to 5 as a facilitated discussion. We'll invite everybody back up here and to answer some questions, and hopefully there'll be good participation. So please stick around this afternoon. I know it's beautiful outside, but you can go for a hike afterwards. So what I'm going to talk about is 3 projects that kind of work in different ways in building collaboration. And the first of those, these are the folks that worked on these projects. I was involved in all of them, but I'm here representing for most of them. And one of the first one I'm going to talk about is promoting coastal resilience and adaptation in coastal Arctic Alaska, mostly in the Bering Sea, Western Alaska region, the Aleutians. And that is a project that really kind of works on just kind of collaborative learning. And then following that, I'll be talking about some work that we did in Southeast Alaska that's really about collaborative planning, and then I'll go into an actual adaptation plan that we're working on with the Nome Eskimo community that is actually about action. So this is kind of a timeline or a scope here of ways you can move from just learning about what's going on in your area and the changing climate to actually taking action on doing something to help build resilience and adaptation. So the first project is really sponsored by a whole host of groups. This is a big effort that we had lots of different workshops that I'll talk about, but you can see all the logos up there of the representatives. We had over 230 participants in this project that helped make the materials for this. And this was led by the Landscape Conservation Cooperatives. If you're not familiar with them, they are a public-private partnership. The ones that we worked with the most were the Aleutian Bering Sea Islands and the Western Alaska, what we call LCCs, not LCs, but LCCs. They're largely funded by the U.S. Fish and Wildlife Service, and their job really is to bring people together and collaborate. And so I've worked with them over the last year on one of these projects, and we're continuing to build that collaboration. And we started with doing some coastal resilience workshops in Western Alaska. You can see images here from some of those. And our work was to just really provide information to communities. This is the real basic type of grassroots efforts to try to just get communities information they need to make informed decisions and help them have a voice in what's going on in their area, bringing some of the latest climate science to them, and then trying to coordinate those different efforts. So it's about information and tools they need to better respond to coastal climate change challenges, and then help them support healthy habitats and resilient communities. So we had we brought out lots of different speakers from the Alaska Climate Science Center, from NOAA, from Sea Grant, and others to kind of talk about what's going on in their region, and then have a dialogue about some of the local responses. And the goal here is to provide an opportunity for decision makers, resource managers, and environmental agency professionals and communities to work together to kind of facilitate some kind of action. And these are the 4 places we chose. We're trying to get together some funding to actually do Bethel as well. And so these started in May and then ended up in December up in Kodiak. It's a great place to visit in December. It's cold there. So the methods are a 2-way kind of information exchange, but the big part is to actually build a network and then provide people with some tools. And so this was kind of the outcome of this, was a summary of coastal climate change and then what to expect in the future. We call these the toolboxes. And they're these really long documents that will be available in paper and electronic formats to help describe what's going on. They're very simple. They're just a couple pages for each different topic. and they always end with what can you do? And so there's lots of different things in here, from understanding coastal erosion to actually building greenhouses and food growing infrastructure in your community. And examples of what other people have done and who to contact if you want more information. We also put together these really cool posters, and these posters have over 200 different authors. And you can see people working on the posters. We provided a basic template, and then we let everybody work on them. And the end product is actually quite striking. We had Agnew-Beck, which is a planning organization, facilitator in Anchorage, kind of help us with this. They have the Center for Environmental Visualization at the University of Washington put these together for us, so we kind of had to give them a blank canvas. We're also in the process of maybe removing some text from it and making them more visual, and then eventually they actually, this summer and next fall, will become electronic format, so I'll talk about that at the end. So you can see one, this is one. We also try to give them nice titles like The Ocean is Our Grocery Store, things like that. And so this one is about the changing coastline. And then of course, when you start talking about climate change in a rural community, immediately everybody talks about subsistence harvests. And so this one is about how the climate is impacting people's subsistence way of life. And ultimately, what we like to do is get these out into communities, but we'd also like to get them on the walls of policymakers. Senator Murkowski has actually expressed interest in putting them in her office, so we'll see if we can make that happen. And we'll continue to share out some of these strategies. And so this will be published here soon. I have to end with, this was a huge effort, so there's a lot of authors on these. It's led by the Aleutian Pro-Law Violence Association, the LCCs, Agnew-Beck and Amy Holman from NOAA. So it's a big effort to put all this together, and we're gonna get together at the end of May and have kind of what we're calling the All-Stars Workshop, all the people from all these different efforts coming together and talking about what we should do next. So the second one is moving from giving people information to actually doing something, to planning. And this was a 2-day session that we had at the Southeast Alaska Environmental Conference in Southeast Alaska. The Central Council of Kodiak Indian Tribes, or environmental program, does a great thing where they get together all the tribes once a year and they talk about some of the biggest issues. And they decided that year to tackle water and climate change planning. And so they had already gone through a process of identifying what was most important to them. So I worked with Ray Paddock at Central Council and Chris Whitehead at the Sitka Tribe to put together a couple-of-day workshop to talk about this. And so we collaboratively led a session, and there were about 80 participants there, about 60 participants from tribes at any one time, and about 20 people from other agencies, the university. I brought in a lot of people from the University of Alaska Southeast who are doing some of the latest and greatest climate science work in Southeast Alaska. We talked about mitigation and monitoring activities, what could actually be done. I gave them questions like, if you had no money, what could your organization do today? And if we gave you $1 million, what could you do? So, helping people think about what they could actually do in their communities, just to start very simple things like monitoring activities. Then we reviewed some of what's going on, and then at the end of this, the best part of this was actual partnerships coming out of that, facilitating that collaboration. And these are the things that they decided to work on. They decided not to tackle climate change in entirety, but think about some of those resources that are most important to them. Salmon, shellfish, berries, yellow cedar, and then cultural sites that may be being impacted by coastal storms and other things, and then also human health. And one of the key things is to actually bring together not just the environmental coordinators from each tribe, there were 17 tribes in attendance, but also bring in university researchers, NGOs that work in the area. Southeast is blessed with lots of non-governmental organizations that work on the environment. state and federal, the U.S. Forest Service was there. They sent 4 people, which was really great to have the largest landowner in Southeast Alaska at the table, saying, hey, we can provide these resources and services here to tribes, and we can work together collaboratively to do monitoring type of activities. And then the North Pacific LCC helped fund this, so we brought in that big key partner. And then, of course, the thing that I want to really get across here is that the tribal environmental coordinators were the local experts. These are the people that are actually going to work in each of the communities, put out some of the monitoring equipment, and do that type of work. And they've also become some of the key leaders, especially in Southeast. And Chris Whitehead's here. He's going to talk about his program. And I think that's a really interesting case in how a tribe can actually take this research on its own and then work with partners to make it successful. We didn't want to tackle, one of the things that I've always been bothered by in terms of climate change is when people come in and talk about projections, and they'll give you 100, 200, 300 years into the future what your landscape's going to look like, and it's all doom and gloom, and everybody walks away depressed. And what we tried to think about was, think about the next 2 or 3 generations. What will the landscape look like for your children and grandchildren? Let's just think about that. A lot of the infrastructure in communities in rural Alaska were built in the 1960s, and it's aging. It's infrastructure that needs to be replaced and repaired. And so as we move forward to build for the next 2 or 3 generations, what can we do to build communities in a better way of life for people, for our children and grandchildren who are going to live, or hopefully live, in those communities? So think about that. And then we talked about telling stories. This is an early iteration of our poster. So you saw the beautiful version with all the nice graphics. This is the one that people actually got to work with and use narrative to tell stories about that. So lastly, I wanted to just talk very quickly about another project we're doing in Nome. And this is kind of like the finals case. And that is actually taking, not just talking about what's going on in your environment, and then not just talking about those things and then planning for monitoring activities, but actually putting a plan in place. How do you do that? So I worked with Nathan Kettle from the Alaska Center for Climate Assessment Policy, and a couple of people from the Nome Eskimo community, and we did a plan for the community of Nome. And Nome itself is kind of a broader scope. There are actually 4 communities there that you can see, 4 tribes that worked on this together. And the idea was to craft a plan, an adaptation plan for them for the future, and to think about those things that's important to them, but also what's reasonable to do. And so this was our team. You can see 2 people from the Nome Eskimo community that were engaged in the process. They really drove this process. We were just there as support staff to help write the plan and take all the information and all the things they wanted to do and put it together for them and work on them collaboratively in that process. And so we had some scoping interviews. Nathan went out and interviewed people, and then John Walsh from the International Arctic Research Center put together a presentation. He basically took all of the information Nathan gathered from local people and said, this is what people are observing. And then he took what the latest climate science is saying, put them together, and guess what? It all matched. So we did some workshops after that to talk to people about those things. And the things that people were concerned about, of course, are food security, subsistence, their erosion and impacts on infrastructure, and shipping in the area as the Bering Sea becomes open to shipping traffic. And to adapt, there were very simple things, like the change in weather patterns is impacting people's ability to dry their fish properly. How do you adapt your fish drying techniques to a changing climate? And so they talked about how they could do workshops and get people to work together to figure out the best practices, because everybody's got a unique way to dry their fish or process their fish, and their way is always the best, of course. And so get people together and try to figure out best practices for that. Another one was really simple. There's cemetery that actually belongs to the Nome Eskimo community. They own the graves on the cemetery, but the land actually belongs to the port and the city. And so how do you get those two to work together? And then there's also some subsurface rights that belong to the local native organization. So how do you get all of these partners into the room to actually deal with this? Because the erosion is taking the graves away. And these are graves that were a part of a 1918 epidemic, and there were other later influenza and smallpox victims in there. And you do not want these things to be open. You don't want them eroding. So really it's a matter of how do you get people to work together to deal with this issue. It was very simple. You know, there's not a whole lot of infrastructure work that needs to be done. But you know, it's a matter of getting everybody into the room together, and that was an outcome of this that was really kind of easy, tangible type of thing to take on. So finally, I just wanted to talk a little bit about the process of putting together projects like this. And you know, the goal is really to build a network of collaborators. So whenever you're thinking about community-based monitoring or having communities help you with your research, it's including them at the very beginning and talking to them about what's most important and what would they like to see as a final desired outcome or product of this. And then pull together, you know, everything that's going on, work with the people that are doing the latest research in your region or area, put together a synthesis, and then together collaborate and build a plan for moving forward and how to take action. So as examples of outcomes of all of these efforts, recently NOAA had a coastal resilience grant program, and thankfully it was with FY17 money that's already existing, because we have no idea what the future will bring for coastal resilience or NOAA or sea grants. So it's likely that there's money already there. So we had a lot of grants come out of Alaska, and Alaska's never gotten one of these. And we're hopeful that it's our turn finally. So the governor wrote us a very nice letter of support, as well as all the other federal agencies that work in Alaska. And we put together a coalition for 3 different projects. The first one is called Leveling Up, Building Coastal Flood Guidance for Alaska Communities. Almost every state in the United States has coastal inundation information, but Alaska doesn't. And the work is being done by the DNR's Division of Geological and Geophysical Services. They're the ones doing the work, things that NOAA should be doing. So we put together a project, a collaboration between Sea Grant and that department of the state. And we included 3 native organizations in that, because we want to make sure that whatever tools we're building work for them and are easy for them to use. And also speak to what their biggest issues are in terms of coastal inundation. So we included the Bristol Bay Native Association, the Association of Village Council Presidents, which represents all the YK Delta communities, huge number of communities in that area, as well as Kodiak in Nome, that represents the North and South region. We also worked with the village of St. Paul, and they put together a project. They have something called Bearing Watch. They have a project they put together called Establishing a Coalition of Communities and Agencies in Defense of Alaska's Coasts. And it's part of their Bearing Watch program, which provides a platform for communities to actually collect their own data and then download their own data. So this was a huge collaboration that was partly an effort of this work that we did in the Bering Sea. So bringing together all those people involved in these workshops to put together a proposal. And finally, there was one for Southeast, building community-based coastal resilience in Southeast Alaska, that was headed by the Nature Conservancy, was a big participant in our workshop in Southeast, and a lot of the people that showed up for that workshop put in together to try to put together also a project for this as well. So the point here is that these are projects that are being built out of these collaborations and trying to include communities and tribes at the very onset of a project instead of just having them come in later and help collect some of the data. So finally, I did want to mention that we are, as an outcome, we wanted to build a place for community resilience efforts in Alaska on the web. And that's going to be called Adapt Alaska. It'll be hosted by Sea Grants. It's being built right now by Maggie Beck and has a lot of funding from the LCC program and others. And it will be a location to tell stories about efforts that communities are doing, to share some of the best information, and it will act as a portal to link to all the different sites that are useful for people doing work here in Alaska. So hopefully that will come out as a draft in May. We'll try to get it out by next fall. So thank you for your time. And I will listen and hopefully come up with some intelligent things to say during the discussion section.
Thank you, Gavin. And we do have time for a few questions now before we move on to our next presentation. But in addition, we'll have an hour-long facilitated discussion at the end of the day. So does anybody have any questions? Julie?
So, in terms of adaptation, it seems as if with climate change that it's more focused on erosion, sea level rise, and those kind of issues. Knowing that we're a large fishing community, and that the fishing community needs to adapt to whatever's going to happen in our waters, how could you adapt what you're suggesting here to think about commercial fishing and processing and how the different participants in the fishery have access?
So I think that there's some really good work being done that Danielle will present tomorrow, and that's on the grain of the fleet, and they've looked at how the fishery is changing. But you know, part of this is also understanding ocean conditions, and the College of Fisheries and Ocean Science has been doing a lot of research on changing ocean conditions, especially in the Gulf and the Bering Sea. And so I think working collaboratively with the researchers doing that, but then also working with those like Danielle and Courtney Carruthers and others who are dealing with the kind of the human side of that is something that needs to take place together. And the CFOS, the College of Fisheries and Ocean Science, does have an emerging marine policy type of field that's coming along, and I think it's becoming much more important because we need to integrate all of those different aspects together to try to understand how to adapt our fisheries. That's a great question.
Julie?
Yeah, and I hope something that can come out in our discussion is kind of a follow-up to Julie's question. Is I'm sitting here thinking, oh my gosh, like who are the partners here in Kodiak? You know, we have such a diverse community, not just a diverse fishing fleet, but we have a diverse community with sometimes people with different perspectives than what we might think. Or it's pretty big. I mean, even if you just put our borough and our city council members together, that's big enough and controversial enough. And then you add all these other layers. But yeah, I hope that you can help guide us and kind of form these networks. We don't have to solve the world today, but I just see our community as quite complicated. Not that others aren't.
Thank you, Gavin. Yeah, I was thinking along those same lines. It'd be interesting to kind of discuss further how you bring the collaborative process into determining what you want to see for your community, like encompassing the hub of Kodiak and our 6 rural communities around which you have experience with dealing in the communities out in western Alaska. So. Okay, well, thank you very much. Okay, our next speaker is Marilyn Sigmund with the University of Alaska Fairbanks in Maryland, and she's going to be sharing with us today a presentation entitled, What are the Best Practices for Community-Based Monitoring of Alaska's Coastal and Ocean Environment? Great, thanks.
All right, well, good morning, and I guess I'm a little bit eating lunch, but I'm very glad to be here in beautiful Kodiak and beautiful weather. This is just outstanding. And I'm going to report a little bit of work that we did, what I call BD, before we had David as our community resilience specialist. We're really fortunate to have him. And and going to hopefully frame some of the thinking you might be doing as you're hearing some of the presentations this afternoon, and stimulate thinking about what could be done in Kodiak in terms of planning and even some monitoring collaboratively. So, and I want to, this was a question that the Alaska Sea Grant, who I work with, and the Alaska Ocean Surfing System, who you're also going to hear from later this afternoon, kind of posed a few years ago before the last CAMs to say, they were beginning to see a lot of proposals. They're both grantors for research, and they were getting proposals from both scientists and community members that involved community collection of data. And they asked, well, we don't know, what are the criteria for a successful project? What do we think is going to work? What are the ingredients of a project that will be sustainable and also yield some very valid scientific data? So they decided to hold a workshop, a statewide workshop in Anchorage in 2014. And I did a little bit of background work to figure out, well, what is going on out there? And we came up with the term community-based monitoring, and I want to give you a little context for why we chose that term rather than citizen science, which also was going on in Alaska. So I don't think it's any surprise to anyone that monitoring the environment, environmental monitoring has been going on for 7,500 years, at least on Kodiak, that we have this long-term, people didn't survive unless they had a really keen and often very localized knowledge of their environment and some of the trends and how, what they could expect in terms of seasons and where the animals were and those kinds of things. And they had a very good system for passing it down through storytelling across generations. So we're very fortunate in Alaska that we have this body of traditional knowledge, which is ongoing. And as you heard, and you'll hear from later today, the tribes have taken a leadership in this environmental monitoring and have the ability to bring some of that in side by side with what Western science can collect in terms of data about the environment. And citizen science actually is not new to Alaska either. Julie Parrish mentioned that the Christmas bird count started in Central Park, New York City in 1900. Well, it actually started in Alaska in 1941. The first bird count was in Anchorage and Mountain Village in 1941. So we have, and it's been continuous, at least in Anchorage, so we have 75 years of that type of data. And we have more than 1,000 people, volunteers that go out and do the Christmas bird count. Now the Christmas bird count didn't start, I don't think it's, as she said, it started, we went and shot birds and then counted them. But birders are a competitive lot. And so they really, a lot of them enjoy just trying to get the most birds, or the rarest birds, or whatever. But it has proven to be a really important data set. When Dr. Shannon Atkinson spoke at the Marine Science Symposium about the murre die-off, she said this data set helped her figure out how abnormal that was, because the murres were inshore. And she could go back into all those bird counts and say, when in the coastal communities did they see a lot of murres inshore? And that was her baseline. So even though it wasn't started with an idea, it's going to be a baseline. It actually has proved to be a very valuable one. So what I see is there was a big shift in this kind of attitudes in Alaska about environmental monitoring. There were a number, and when I first came to Alaska in the mid-70s, I worked for a Department of Fish and Game as a habitat biologist. And monitoring in that context was mitigation monitoring if there was a development project. And you heard about one of those yesterday about the runway extension. And it was something happened, a very discrete event, and it was permitted by a federal or state agency. And we always tried to get some monitoring and rarely succeeded for restoration and mitigation. But then we had the 1989, we had the Exxon Valdez oil spill, which was a regional scale toxic pollution event. And in the mid-1990s, these scientists that's when they put together the information to see that there had been this regime shift that began actually in the late 1970s throughout the Gulf of Alaska. It took that long to propagate through the system. And then there was more attention to this kind of retrospective understanding about the Pacific Decadal Oscillation that had been, the environment had been going through a pattern of change that could stretch back to 1900, that warm periods and cold periods. And then now we have what's showing up with this trend of continuous warming, and in the Gulf, what Terry Johnson referred to as these anomalies, but the trend is warming. So the big shift that I think I see is that certainly the oil spill among anyone involved in cleanup and restoration really got the point about the importance of baselines. So they were missing in a lot of situations and had to be either reconstructed or just were missing. And communities got more interested in being involved in monitoring, partly because of the spill, but also because of these large integrated ecosystem monitoring programs that the North Pacific Research Board started funding and brought in citizens as advisors and in terms of talking about the priorities and impact, and what the impacts on hydrotrophic levels that people really depended on. And I think also there was a growing awareness as people started saying that we are concerned about clean air and water, that we have a really limited government capacity, like state capacity, to do some of this monitoring work in our huge state. So I think, it's interesting to me, some of the biggest programs in terms of volunteer programs were in areas of water quality. Now it's growing in PSPs and harmful algal blooms, and also marine debris. And those are all responsibilities of the Department of Environmental Conservation, but there's no way in the world they can be everywhere in the state. And so community groups and tribes have actually filled this gap in the state in kind of a more of a citizen monitoring kind of way, citizen science kind of way, using volunteers. And then because of climate change impacts, which are twice the rate of what's happening in lower latitudes, there's this whole alphabet soup of what they're calling observing networks now in the Arctic. There's the Arctic Observing Network, AON, there's SEON, Sustaining Arctic Observing Networks. There's a couple in the Bering Sea that are international with counterparts in Kamchatka and Siberia. And so because there are people in the Arctic, they're being impacted. People, and we have people who are out on the land hunting and fishing and doing subsistence and berry picking. There's been this kind of natural evolution of bringing their observations and their traditional knowledge into the system, trying to track what's going on and help them think about adaptation. And then there's been some statewide networks that developed, and they're at various scales of rigor. So we have the LEO network, the Local Environmental Observers Network, which is fairly anecdotal, what scientists would say, where they've actually vet their observations because make sure the actual observation is validated within the community. But they're kind of more opportunistic observations of unusual events, a species out of range, disease in a subsistence species. And that's been this huge network in many, many coastal communities is share information with experts who can help them interpret the significance of it statewide. And at the other end of the scale, we have the Alaska Ocean Observing System, which is this aggregator of all the scientific data that is coastal. And I think I was reading what they have, something like 3,200 different sensors out there. They're now feeding their data in real-time sensors out throughout Alaska's oceans and along the coast that are feeding into that system. And then programs like Both Watch Alaska and GOER all feed their data into this system. So there's a huge amount of real-time and near real-time data and even historical data that's accessible scientific data. So you get this feeling, we get this feeling, there's just all these people out there looking around, sharing information, seeing interesting things, trying to figure out what they're seeing. But Sea Grant said, okay, let's try to get our arms around this. We know that we're a funder. There's scientists and engineers out there who are looking to work with community members and vice versa. And the question for the workshop was, what are the best practices for success? And what we found in kind of doing our homework about what was going on is that what was going on in Alaska really is unique. It wasn't as the classic citizen science where a scientist like Julia Parrish basically creates the project and creates a way that people can be trained to collect very valid data, and they have a way to vet that data, and then they do report back to the people about what they found. But it's really driven by the scientist. We found that all kinds of communities are being engaged in this kind of monitoring, from the birders to the geographic communities to people with just, I call them affinity groups, people who like to walk beaches, people who like to pick berry subsistence users. So a lot of diverse communities engaged. The intensity of engagement is more than just data collection, and I'll talk about that in just a minute. Some of them are doing monitoring over huge geographic areas. There's a diversity in what is being monitored, and there's a diversity of methods. So what we found is there's kind of a spectrum of community engagement and collaboration. And so all the way from a community member or community government or organization or a tribe saying, we need to monitor this. And we've done the planning to figure out we have to monitor this. They're engaged in project design, data collection, interpretation, response to results. They can be engaged in every single stage, as David mentioned, of the process. Whereas I think with citizen science, the focus is more on the data collection piece for the community members. But these projects and the people that came to this workshop talked about being involved in all parts of it. And this is just kind of a snapshot of the kinds of things that Alaskans monitor: dead birds, of course, harmful algal blooms through plankton tows, subsistence biomonitoring. A lot of the organs from some of the harvest marine mammals are analyzed for contaminants and things like that, and condition is reported. There's invasive species monitoring like crab, green crab, PSP, digging clams, and your standard water quality, and maybe have some things called bioblitzes. I think Kodiak might have had a bioblitz for biodiversity. And of course, marine debris, not only just to collect it and get it off the beaches, but to analyze where it's coming from and try to figure out if there's a way to prevent it. So this is just probably a partial list of all the different kinds of things that people do monitor in Alaska as community members. I don't want to call them non-scientists, because I think everyone is a scientist in their own way. But they are monitored using standardized protocols so that the data that are being reported have some validity and ways of being managed to check the quality. All of these, there's a program for every single one of these. And I put a star by some of them because they're kind of sentinel, sort of sentinel programs where the purpose of the monitoring is if something is detected, like a huge die-off of birds or a PSP at a certain level, it triggers something in one of the agencies that's responsible for dealing with that. So we came up with this definition of community-based monitoring, broader than just citizen science. It's basically a collaborative effort, and it involves community members who do collect observations and data, but it also involves some sort of local organizational partner or partners, and some that are even beyond the community, who do support the data collection, data management, data use in various ways. So there's a partnership there, and some of it's all in the community, but some is organizational and some of it's individuals, some of it's the community and agencies that are state and federal or beyond that. And we kind of follow the lead of one of the UN groups working on conservation in the Arctic, because they also termed what was happening community-based monitoring, and they said it was a combination of sensors like instruments and people as sensors. And those are the kind of systems we're seeing all over Alaska. So April 1st and 2nd, April Fool's Day, 2014, we had this workshop. We had people from all over the state, come from 32 different communities and from 7 other states in Canada and Russia. We did a lot of sticky noting and discussion and presentations. Julia Parrish was one of our keynoters about citizen science programs. And we had a lot of sponsors, some of the same names that you saw when David also put these names up. These people are continuing to work in their regions in terms of this type of planning and monitoring. We also did a Wordle out of that, and the big emphasis for everyone was on community and data, but you also see people, program, and of course, funding. And so here's just an example of, there's a lot of agreement on what people said they wanted to see come out of these programs for the communities, and for the scientists it was a little bit different often. So everyone wanted to see consistent collection of scientifically defensible data. They wanted to see baselines that they could establish, so then later they could determine if a significant change had occurred. And some also mentioned, if a baseline is what you want, you also need to know when you're done. So that was an interesting message from people who said have an exit strategy if you want a baseline. They did want to be able to have community-based data that would trigger a government. They wanted to actually see the government respond. If they were on these sentinel programs and they had a problem, they thought it was success if then they got a response to what their data showed, as well as being able to take that data and immediately give it to the community to support response to environmental change and emergency preparedness. And this is one of, I think, the real messages I got was this process shortens the traditional scientific method of collecting data, publishing in a peer-reviewed journal, having someone like myself who's more of a science educator try to translate it and then be useful to a community. This is direct and short circuit to get the data back to the community and so they can use it. And then one of the really important things that came out of this type of work is it's a success if you can sustain your partnerships, and that takes a whole lot of trust, and you build trust over time. So I feel like it's a little bit of an infomercial on a talk show, like buy the book, read the book. But this was the product of this workshop. It's kind of a how-to manual, and we did it in a format of a checklist that you can go through if you're planning a project or trying to improve one. And it quickly leads you to, if you're going to do this in your project, here's a paragraph or two of the best advice from all these people that have learned a lot of lessons by doing these kind of projects, so you can ignore the rest if it doesn't describe your project. It's also fairly handy if you're trying to write a grant proposal and you want to see that you're addressing everything that a grantor might want to know about your project to see if they think it's going to be successful. So what we heard, I'll just give you a few highlights from this. I do urge you, there's copies outside if you'd like one. And you can also download it for free as a PDF from the Sea Grant bookstore online. But we heard that success in community-based monitoring requires that you do come up with some common goals and objectives. That also at the outset, as David mentioned, you clearly define the expectations on both sides, on either the scientist side or agency side, as well as the community side, so that's how you start building that trust, and later you can evaluate whether your expectations were met or something needs to be tweaked and adapted. And you do need, so defining success at the outset is important, as well as what are the metrics of that success. But as I mentioned just a minute ago, scientists and community participants often have different definitions of success. So scientists are pretty straightforward. They need valid data, and if they're definitely specific, particularly if they're academic, they need publications, peer-reviewed publications. But even if they're an agency scientist or a program manager, they need to have results that they can use to meet their agency mandates for protecting clean water, clean air, whatever their mandate is, land management. Whereas many community members may be looking more to their own engagement and feeling some reward for being engaged. And often that's knowing that they're making a difference. And so that's where there's kind of a tension between what kind of methods you use in terms of them being scientifically valid and what's really feasible for your participants to do well and do at a valid rate. So there's a lot of discussion usually around that. For someone who is a subsistence hunter who might be participating by providing samples from their harvest, they're bigger, the reason they often would be engaged is they want to hopefully be able to sustain their harvests, and communities often are involved in how do we sustain our resources. And then there's a lot of involvement with often students and teachers, K-12. And for any of you who worked in that environment, it's a challenging environment to work in because you have to make it as academically enriching and meeting the standards or curriculum framework of that school, and teachers are really pressed for time. I had a conversation about that just last night. So, but fortunately, in the next generation science standards, the new national science standards, there's a huge emphasis on getting kids doing real science. And so, there's a lot of interest in participating in these projects, but it takes a lot of work to tailor it to where it can be done by students. We found that there were just some really common challenges to all of these programs. The first I mentioned was developing these partnerships and sustaining them. Also developing these data collection methods that are scientifically valid on the one hand, but feasible. And then data management. I'm hoping that the person from AUS will talk a little bit more about this. There's a lot of conversation about how you do the quality assurance, quality control on your protocols, the training, that sort of thing, and then who's going to manage the data for the long term, because some of these data sets are going to be valuable far into the future. And big emphasis on communication, and what, this is a new term for me, the co-production of knowledge. And that was, I talked with Carolina Behe with the Inuit Circumpolar Council, and she said, and I'm guilty of always, in the past, saying, we want to integrate Western science and traditional knowledge. We want to translate traditional knowledge into Western science. And she really chastised me a bit and said, what you have are these two different knowledge systems. And they're coming, they have different methods of collecting their knowledge and transmitting it. And what you really want to do is put them side by side, respect both of them, and co-produce the knowledge that you have. And I thought that was a really good way to think about it. And there's a lot of advice in this manual about ways of doing that and guidelines that come from the Inuit Circumpolar Council. And one of the things we heard from almost every community is they really want to engage their youth. And that is the challenge because of some of the things I mentioned with K-12 classrooms. But there's also after-school programs and summer camps. And of course, funding, and I have to say, this was in our 2014, that was certainly not the challenge it is going to be in years to come. That's probably risen to the top as a challenge in our current federal funding environment and state budget cuts. So, so we have, I want to mention the resources that you have as a community or an area to develop your own programs or grow the things you have already going on. You can download this manual from our workshop. We also have from our website. We also have, there's another website that AVES hosts called Alaska Community-Based Monitoring, which has many of the presentations that happen at the workshop and other resources, and especially ones to help you evaluate the success of your program. And I just wanted to, I'll also end like Julia to say what you can do. Well, you can volunteer as a citizen scientist to collect data. She mentioned that there's already some things you can volunteer to right now. You can also kind of choose among a variety of ways to participate in a community-based monitoring program. You're going to hear about some of those this afternoon that are going on in this area. You can start your own using this manual, handy-dandy manual. But I really hope you'll all, at the end of the day, participate in a conversation about what is important to monitor in this area and how might you collaborate. That's hopefully what can happen today. I'm unfortunately not going to be able to join you because the last thing Julia said was you can march for science, and I'm actually going to go to Washington, D.C. and march for science and science education. So I have to leave before the conversation. And then I want to have the last few words of advice. Fran Ulmer kind of did the closing notes of that workshop, and she listened for 2 days and said, here's what I heard. Communicate, communicate, communicate. That was the first thing she's talked about because that was the key to all of this, was everyone communicating with one another and building that trust. And then another comment by Sue Mager from Kodiak said, be sure to celebrate your success. So have lots of parties and gatherings and bring your community together. That's a really important part of this whole process. And Julia Pierce had a great zinger that all of us wrote down and put into the manual. She said, be fearless, and she was talking about asking for money, and be kind. That was her. So with that,
Thank you very much. We have time for a few questions here. Anybody have anything they would like to ask at this moment, or you can save them for the end of the day for the community discussion as well? I think they're saving them. Well, we won't be here though, so and
read the book.
Read the book, yeah. There's the opportunity to march in Kodiak as well at 10 o'clock down at the AC if anybody wants to march for science in our community.
I guess, I'm sorry, I do have a question, Marilyn, because you are disappearing. I didn't realize you're taking up. Well, you recognize that funding is a challenge, and the question I jotted down as you were speaking, because you had mentioned clearly define expectations of your project. But, so for instance, the few projects that I've been involved in and initiated are grant funded, so it's very hard to define expectations when you're working on these little short-term pieces. Well, like you can't look broader, like it's taken the scrambling to get there. It's hard to define expectations without the long-term funding.
Yeah, it's very challenging. And I think one of the big differences that I meant to mention in Alaska, I mean, the citizen science model is that there's someone who initiates that, someone who has the funding to do the data management, development protocols, the training, and communications, often grant funded, and everyone else volunteers their time. In Alaska, that really is not the case in many communities. If you have a subsistence hunter who you're asking to help, that's not exactly a volunteer. A lot of stipends are paid, and we fortunately have had this great tribal environmental professional network throughout Alaska that EPA funds, so that's going to be a challenge. And they often are, they're paid people in government whose job it is to do this kind of planning and monitoring, the borough governments and the tribes. So it's going to be a huge challenge if we do in fact lose some federal funding. But the importance of doing what can be done as a volunteer becomes that much more important. And so I think we're going to have to we're going to come to that challenge really soon, hopefully not this year, but soon. So yeah, that's just going to be the reality that if the community understands through the process they go through that this is really important data for them to figure out how to adapt or how to, like the fisheries or whatever it is, then it's going to have to rely, I think, a lot more on volunteer participation, civic participation.
Thank you, Marilyn. Well, Marilyn touched on the value of youth engagement, and I'm very pleased that our next presenters include some youth. And so we have with us Mr. Serafin McGann. He's the World Bridge teacher, and Kia Shin, Daniel Johnson, Tiger Oka, which are students with the Kodiak High School. And they're going to be presenting on a World Bridge Kodiak Island School Borough District project activities. And welcome, and thank you for being the youth that we can count on for the future. And I'm hoping you can expand on some of the awards that these students have won in addition to the presentation. So come on up. Thank you.
Thank you very much. Hello. Sorry, a little bit nervous, not used to doing these presentations. Thanks for having us, everybody. I'll just start out with a brief intro. I know we've got a very limited amount of time, and we're standing between everybody and lunch. So that's a dangerous place to stand. I realize that. So my name is Serafin McGann. I was hired by the Kodiak Island Borough School District this fall as a classroom teacher. I've been there since January as a shop aide, where I was helping out with the wonderful CTE program at the Kodiak High School. If you haven't gone over there to visit it, you should. It's very, it's inspiring. Anyway, I teach robotics programming, and I also work with a program called World Bridge. And this is a pretty interesting program. In a nutshell, we're basically building an R&D lab at the high school level, which is something you probably see more at like the university or in a corporate or governmental setting. Again, please feel free to come visit our lab. It's F104 at the high school. Just come by. I'd love to give you a tour, and our students can show you what they're working on. And so, basically, it starts with the students. There's a few shots here of some students in Kodiak on the left. We've got some, we've got some pretty, decent capabilities. We've got a nice solder station. I'm trying to upgrade it so we can do micro soldering. We've got a 3D printer, a laser engraver, sort of what you would expect in like a maker space type of environment. And there's one of, hopefully, a future student driving or flying one of our drones, drone practice drones. And then on the bottom right, there's a shot of some students at Phaeton Academy in Middleton, New York, because this is not just a local, something local to Kodiak. We actually work with schools in other parts of Alaska and around the country and in other parts of the world. And then this is just a shot of actually our old lab at the Fisheries Technology Center. I believe I probably didn't get that quite right. I'm sorry, a little nervous. Anyway, this is just a shot of our satellite tracking project and two of the higher-end drones that we have. These are, professional level. They can take, 4K imagery. You can put, they can lift about 40 pounds, something like that. So they're very high-end machines. And then we have our partners. So this project, it's about having students do real science. I'm glad we were placed after Miss Salzman? Sigmund, sorry. Miss Sigmund's presentation. It's a really nice dovetail. These are some of the partners that we are working with. And I don't want to eat up any of more time. I'd actually like the students to speak. You're probably tired of hearing me. And so we're going to start with Daniel Johnson, and he's going to tell us a little bit about our earthquake signal precursor project.
Okay, thank you, Mr. Serafin. As you all know, my name is Daniel Johnson, and the first project we'll be talking about today is called ESP, or Earthquake Signal Precursors. We work with various partners around the world with donations or just research, and we work in alignment with the United Nations Sustainable Development Goals of risk reduction in an effort to test the theory that electromagnetic field anomalies precede earthquakes. Right here is a picture of one of our sensors. We have sensors in various locations. One of them is in Kodiak, Copper River, one's in Copper River, Ketchikan, Old Harbor, and there's also a new one in New York, in Baytown Academy. These send data to our servers in our building, and the servers then record the data and then send it to our website, and they're graphed out over here on the website. And if you look up here on the top right, there's a 6-month distribution map of all the earthquakes that's happened between February and July 2014. As you can see, there's thousands and thousands of earthquakes over the 6-month time period, and because we're located on the Ring of Fire, we have over 200 earthquakes per day. And scientists are really interested in our data because we're located so close. And this is one example of one of our earthquake anomaly reports. Oh, yeah. Okay. So, these are produced by A-World Bridge students, and after an earthquake occurs, we're notified via app notification, because we're in the technology age, or email notification from USGS. And then these will tell us the magnitude, the depth, and the exact location, and the exact time of the earthquake. We then make a quick report, then we subtract the latitude and longitude from where our ESP sensors are, and then we record that too. And then we try to backtrack through all of our data, or not all of it, just the time period before it, and we try to find the anomalies. And in this case, we did find anomalies. They were unipolar waves right here, here, and here on the X, Y, and Z axes. They happened 13 hours before the earthquake, and they lasted for about 10 seconds too. And if you look here, at the top right, this is the website that allowed the A-World Bridge team in Kodiak to win the NASA Europa Challenge 2 years in a row, 2015 and 2016. And this website includes graphs of all of our sensors. They include vector directions of the magnetic field, one here in Kodiak you can see, and one here in Kenny Lake that you can see. There's more, but the Web World wind map is too small to see them all. And if you look over here, these graphs are interactable. If you go over here, you can adjust this way or this way, and then you can select the time period for which you want to view the electromagnetic field data. And if you go right here, you can select to average the intervals too, just in case you don't want to see everything, you just want to see a smaller version. And you can also download all of our data from our website. It's linked somewhere on the website, I don't have it up here. And then this is a infrastructure of how a diagram of our infrastructure from our sensors all the way to our front end, which is our website. And I'll have Mr. Seraphin explain more about it.
I'm not going to spend a huge amount of time on this. We just don't have time. If you're interested, more interested, again, just please stop by or give me an email. But basically, this is an example of. So this is an example of like a full stack from sensor data over here at this end coming into our magnetometers, through the platforms that we built, through the firmware that we have on our sensor nodes, and then it's transferred into our database server, and then from the database server it is served up on our web front end, and we store it in a time series database, and we also serve it up for in bigger chunks for scientists who want to analyze it. This is all pretty prototype, but it kind of just demos that students can build pretty complex infrastructure, and like this could be applied to pretty much any type of a sensor platform, what have you. Kind of interesting to talk about the community-based data monitoring. This is more or less the type of system you're going to build in any data monitoring application. So it's neat that we can kind of do it at the high school level. Anyway, that's probably about all I want to go into that. If you've got questions, we'll have some time at the end. And we'll show actually one more thing here. Okay, and this is just a slide about just sort of this idea that we're doing R&D at the high school level, and it seems like it would be a good fit for other communities that are doing projects that involve data, because we are solving these problems, as like many others have, but we are solving all these problems that we're talking about. How do you gather, store, how do you maintain all of this data? We've currently collected about 100 gigabytes of data, so it's getting pretty big. So we are actively working on all these problems. And this is a picture of the trophy we won from the Europa Challenge, and we're hoping to compete this year as well. And I think at this point, let's introduce Kie Shin, who's going to tell us a little bit about our NanoAg project. Okay.
Hello, I'm Kie Shin, and I will introduce our agriculture project. So Kodiak is 100% renewable with the energy, but not the food, because majority of food comes from outside of Kodiak. One of the reasons why there are no farms in Kodiak, no major farms in Kodiak, is because of the massive amount of compressed volcanic ash under the soil from volcanic events over 100 years ago. Now this volcanic ash under the soil makes soil into very acidic condition, which obstructs plants' growth. And we will be testing effectiveness of nano-agriculture to create healthy food, which may also use at our school cafeteria. And construction of the greenhouse has just finished. We're conducting new experiments in the hoop house to demonstrate more plants. And Tiger will be showing interaction between UAS project and the nano-agriculture.
Hello, my name is Tiger Oka, and here, last year we were asked by Mr. Berries to if we can fix the golf course, the Bear Valley Golf Course, and so in order to know our treatment sites, we use our drones guided by GPS waypoints to take high-resolution geo-reference pictures of the golf course. Then we stitched them together using drone deploy and made an ultimate map. Because the pictures are made to make the ultimate zig map has its longitude and altitude. We can also make an altitude map. As Kia said, Kodiak's soil was acidic because of volcanic ash from hundreds of years ago, and Kodiak has a rainy environment. All the ash from the higher altitude areas, like such as hills, drain down to the lower areas, causing the soil to become more acidic. And after we made the map, we asked Mr. Berry to show us some locations. We can test nano-ag sites, and we use QGIS software to map locations.
This is one of our treatment area. This picture shows, as shown above, this soil was extremely unhealthy and showed no signs of improvement. The maintenance had fertilized, watered, and seeded this area, but nothing helped the soil quality until our team treated this area. After our team treated this area, an extreme regrowth occurred in the 4 weeks interval as a result for our nano-agriculture treatment, and enabled the existing root system to take over and repair itself. The grass demonstrated strong dark green leaves growing from underground root structures. We have done several soil and water quality tests via burner approach. The collected data was analyzed by our team to determine amount of healthy substances. And our upcoming project is growing several vegetables in different conditions of soil in our hoop house. And Tiger will introduce in the UAS project.
The unmanned aircraft systems operate course and Woodbridge train students so they can operate survey quality drones such as Mantris here, and to take high quality pictures and documentaries for projects such as NAMRAG. And this is a life changer because this can train students for career and professional level skill sets through these operations, and they can also teach. We can also use it for other projects such as construction inspections and fire zone monitoring. Oh yeah. Sorry. A couple months ago, we were asked to do a construction inspection on the Kodiak launch pad and an environmental impact study on the marine life and the wildlife surrounding it.
Sorry. As we progress to the future, cities are experiencing a growth of demand in various resources. City Smart will help facilitate the collaborative efforts in the creation of sustainable cities. In order to expedite the development and creation of sustainable cities around the world, we students have started developing our own City Smart prototype application from Kodiak's 100% renewable energy system using wind and hydro generators. We're also looking into energy production through biofuel and solar energy systems, as well as the water purification.
Okay, and thank you very much, everybody. And this is, we're going to give another short talk tomorrow just about some of our upcoming work and our current projects. And I think, yeah, I think we have a few minutes for questions.
Thank you very much. That was a great presentation, everybody. And I have a lot of hope for the future with our high level of student engagement. And we know our high schools will be funded, so we're good. Do we have any questions for our group here? Julie?
Thank you guys. I'm extremely excited to see you come and present here. I've only heard about a World Bridge. I'm right here in Kodiak, and I'm also a researcher and community educator. And I know very little of your project, so thank you for sharing. It's really exciting to see the level of complexity that you're working with. Can you tell me each your age and how many students are involved in this, and not your age, but maybe your grade level, and maybe how you became involved, or your interest, how you got in?
Okay, so I'm a senior at Kodiak High School. Yeah, makes sense. I'm a senior at Kodiak High School, and I have a friend in World Bridge, and I've always wanted to code or learn how to code. So I learned pretty fast after I started because Mr. Seraphin, our teacher, he's a really good coding teacher. He knows everything. It's amazing.
I'm Kishin, engineering high school. I moved from Korea 2 years ago, and I knew how to code a little bit, and I heard that they have coding class in high school, so I was really interested and I joined, and they had several more interesting projects, so I got involved and learned a lot.
My name is
is Saigoka, and I'm a junior in Kodiak High School, and I was introduced to this program like through TA. In the beginning of the school year, I had an open course, so I wanted to like fill the course. So the Workbridge project had like UA unmanned aircraft systems, and I was interested in robots, so I got involved in it.
My question is, how do you choose your projects? Do you guys reach out to these different groups, or do they come to you?
Well, I think it kind of depends. There's a man, Ron Fortunato, who kind of manages these partnerships. He's contracted by the district, so that's kind of, I don't really work too much on that level. Personally, I would really enjoy to have a lot more local partnerships, because I know there's a lot going on in Alaska, and I'd like to get a lot more involved with the local community in working on these things. And for example, one of the local, we'll talk a little bit about it tomorrow, we're working with Andy Schroeder with Island Trails Network on some projects there. I think there's some overlap. Yeah, and I think there is kind of a mix of both. That's kind of my impression on it. As far as how all that works up at the partner level, it's kind of a little bit above where I'm at. So, yeah. Anyway.
If I had a question as well. You kind of referenced the anomalies and that signal an earthquake, I guess. Could you kind of expand on what the anomalies are and how that can be used in the future, perhaps?
Go back to the slide too. One second. Skip. Hold on a second. Okay, so every time an earthquake occurs, we have a theory, or there is a theory that they send out signals before they happen. So every time they send out a signal, we try to catch it as an anomaly, because we have a baseline of what the electromagnetic field should look like. And so when we see the anomalies, we can record these and keep recording the data so that we can build up more and more data and eventually try to find like an equation or prediction that can help predict earthquakes as they come. So if we see these early on before the earthquake, we can have like a program that finds the anomalies before the earthquakes happen. And then if we can eventually achieve this, we can potentially predict the earthquakes and reduce casualties and property damage as they come.
What's the time difference that you're saying anomaly before?
They vary with each earthquake. In this case, this one occurred 13 hours before the earthquake, but they can occur within a couple days before or a couple hours.
But we're not sure if this anomaly stands for that earthquake, because we have several small earthquakes right nearby the sensor. So this is just our report as what we've seen in our graphing program.
Along those lines, is this maybe, we have an understanding that some of the animals seem to know when an earthquake is occurring as part of this hypothesis that this is how these signals are being interpreted by, say, the ants that head for the hills?
Yeah, I mean, I can just speak a little bit to that. Basically, the idea is that before an earthquake happens, as the Earth is being stressed, for example, ions get released, there's magnetic field anomalies, there's a number of things that happen, and probably that has something to do with the animals being affected, earthquake lights, all of this sort of anomalous phenomenon, like lightning that happens before earthquakes, stuff like this. There's a lot of anomalous phenomena, and the theory is that a lot of that is related to these ion releases. You can see it. One place you can see it is in the ionosphere, where basically these bubbles of positive ions come up and they make the ionosphere a little more rough. So you can see a change in reflectivity over the fault lines before earthquakes happen. Generally those are, I don't know what the time, like I don't know how predictive they are, but I think these things have kind of been observed. So there's a lot of anomalous phenomena, and we were using these IntelliCell sensor stations. But we kind of, the thinking was, this happened before I was a part of the program, but the thinking was the magnetic anomalies are much longer range. Whereas these ion releases, you have to be a lot more like right on top of the fault line to see them. So that's why we kind of went our way when we developed, actually the students, I wasn't even involved at that point. They developed the magnetometer only platform, focusing on that signal, instead of looking also at ions and gas changes and things such as that. But yeah, in a nutshell, yeah, you're exactly right. That's sort of the theory.
That's fascinating. Well, thank you very much. Really appreciate it. Thank you, guys.
Thanks for having us.
Yeah, thank you.
We have this group coming back tomorrow to expand on their work, and it's super exciting to see their work. But let's let everybody out the door. It's really beautiful. I've got three announcements. If you're going to join the Elliptic Museum tour, hit Patrick. We are running a little bit into lunchtime, so I'm going to, Patrick, do you think you guys can get back in an hour, or it's pretty pushing it, or? Yeah, let's at least, let's reconvene at 1:45. We're set to reconvene at 1:40, but let's let a little wiggle room. And for those that are not joining Patrick, there's some reports of whales at the spit right now. Sorry, Patrick. Maybe you guys can walk away of the spit. There's some orca pods that moved in. I encourage you to get some fresh air. Bye, everybody. Be back at 1:45. Thank you.
I've got that text about it. I'm like, I'm not saying anything.
I'm like dying on this.
I'm scheduled to go have lunch in the office. Oh, right there. Yeah. Somebody's holding it. Okay, right after we'll be here, and I'm a little late into it, but 1:40 at the latest. 1:40, yeah. And I'm gonna stay here. Just couldn't help it. I know, it's really actually pretty cool. No, we're not asking for that. It just seems like too much time. We're just going to presenting. I'm going to go to the powerhouse for lunch. Affiliation, name, affiliation. I've only done that with it. And plus we're usually oh, no, yeah. I know, go. If you know of anybody else that you're working with that would be interested in that discussion about community collaboration. I don't know if it's we're having, yeah. I really wanted to put the concept together, but I realize there's a lot of everyone's impressed with what we're
Yeah, I think with the running start.
That's okay. See you for today.
I'll be there tomorrow midnight, Thursday morning.
I know. Well, maybe you can catch someone else. I used to
They're just out here. I do.
I heard that they were in the channel.
In the channel.
You can see them off the spit.
I will head
Let's go watch the killer whales feed.
Okay, go outside to the sun. Thanks, Gordon. No problem. If you see anyone else, send them back in now.
All right.
No excuses. This is a hard
They need
afternoon
a
to.
good excuse.
I know. This would be a hard afternoon to fill. I say maybe we just all go outside and talk. Yeah. Bye, Gordon.
I'm just a couple of hours to go watch the killer whales.
I know, right?
I enjoyed
Yeah, I need to sit. I need to hide over here.
When are you supposed to
Teresa? Man, it's gonna be hard to get people back today.
I know, it's so amazing out. I wish I were outside.
We all just leave. I'm one of the people that wanna go.
Right, I know. Why don't we move it all outside?
I know, and this is gonna be tough because,
I'm not planning to be here tomorrow. I have too many
Right.
other meetings.
Yep.
But I've had some people mention they'd really like to have it streamed because we've
Are
had.
people watching it?
Yeah, they
I
are.
haven't been able to even keep up with the
Yeah.
conversation or.
I
yeah.
haven't. Oh man, there were a couple of things this morning. I was like, oh, I wish I were listening. I couldn't
I
even listen
know,
this
well,
morning.
is it, I never really understood, is it actually recorded too?
On YouTube.
Because it's on,
Yes.
wow.
Yeah. Thank you so much.
I know, I knew, well, once it got to YouTube, I figured it was sticking.
Yeah, we'll just keep it up. We had some glitches yesterday, so it was.
Some pieces missing.
Yeah, in fact, it's still recording right now, so we don't care what we say.
Oh,
No, we don't have, we're in Brooklyn. Anyway.
Conversations yesterday.
Yeah, it's possible.
Well, I don't know for sure, but there was. It just, when did it turn on?
See yesterday.
Oh, wouldn't that be the